Okay, so can you hear me okay now?
Yes, please. Yeah.
Yes, you hear me okay? Okay, I don't hear anything at your end. Can you hear me?
Sir Rich, is it audible? Is my voice audible? Yeah.
Hi, Prash, can you hear me?
Yes, sir. Yes, sir. Your voice is audible. Is my voice audible, please? Yeah. Sir Rich. Sir Rich, is my voice audible, please? Yeah.
Okay, can you hear me?
Yes, please. Yeah. Thank you. Thank you so much. Yeah.
Good, I didn't get that. It was very blurry.
How about now, sir? Is it audible? Yeah.
Yeah, can you hear me? Okay, fine. So I'll talk for about 20 minutes. Okay, so what I want to do today is to talk to you about how basic research is so important in terms of the things that it can lead to. And I will focus on aspects of biomedicine. And I'm going to use some examples from my own work over the last almost 50 years now, long, long time. So I want to start off by talking about the postdoctoral period I spent at Harvard in 1969 to 1972, sequencing tRNAs, or one in particular from Staphylococcus epidermidis. This was a tRNA that transferred glycine into the cell wall of the bacterium, but did not work in protein synthesis. And the question was why? What is different about this tRNA? And so I went to Fred Sanger's lab in order to try to get an answer, and in particular, to learn how to sequence RNAs. And Fred Sanger, of course, is very well known. He won 2 Nobel Prizes, one for protein sequencing and the second one for DNA sequencing. And in fact, he could almost have won a third for RNA sequencing. Anyway, I went to his lab, learned how to sequence RNA, set that up at Harvard, and as a result, I ended up being the first person in the Boston area to get involved in RNA sequencing. A lot of people came to me to learn how to do it, one of whom was Wally Gilbert, shown on the left. He had developed a separate method, a different method for DNA sequencing, and his technician, Alan Maxam, came and worked with me. Now, this led to my getting a job at Cold Spring Harbor, which I'll tell you about in a moment. But something happened in between that was really very important. And that was, I listened to a talk by Dan Nathans. He's the person on the right-hand side of this picture. And he shared the Nobel Prize in 1978 with Werner Aber and Hamilton Smith. And Hamilton Smith had discovered an enzyme in Haemophilus influenza that could cut DNA specifically and became known as the first site-specific restriction enzyme, or the type 2 enzymes, as we call them. Now, these type 2 restriction enzymes operate in the following, well, all the restriction enzymes operate in the following way. They're there to defend the bacterium against bacteriophage, and such that when the bacteriophage introduces its DNA into the bacterium, if the bacterium has a restriction enzyme in there, then it can cut up the DNA, But at the same time, it does not cut the DNA of the bacterial cell because the bacterium separates and changes its own DNA by modifying it using a DNA methylase. And so it puts a site-specific methylation onto the DNA, and this stops the restriction enzyme. And this was really quite important because my first job at Cold Spring Harbor Lab depended on the fact that I could sequence RNA, and Jim Watson had hired me specifically to sequence SV40 DNA. But in fact, because of Dan Nathans and the restriction systems, I actually ended up working on restriction enzymes instead. And this turned out to be incredibly valuable because these enzymes formed the basis of the biotechnology industry. And we now know exactly where the biotech industry has gone in terms of making a lot of companies possible, in terms of allowing recombinant DNA to be made in the first place. And they were just invaluable molecular biology reagents from day one. And in fact, they still are. Now, as soon as we started finding some of these enzymes, I tried to persuade Jim Watson he should form a company and start making these enzymes at Cold Spring Harbor, and then use the profits from this in order to support the research at Cold Spring Harbor. Well, Jim Watson, in his infinite wisdom, decided he didn't want to do that it was kind of dirty to be associated with a company, and also he didn't see how you could make money. I think this was pretty typical of Jim at the time, that he didn't have much imagination when it came to forming companies. We now know, of course, that things are very different. And in fact, New England Biolabs was one of the very first companies to start making reagents for research, and in particular was the first company to make restriction enzymes. The company was founded in 1975 and has been continuously profitable ever since then. It was founded by Don Combe, the gentleman in the bottom left. On the right is a picture from a few years ago of all of the workers in the main building at NEB. The company is privately held and has been that way, and deliberately so, because one of the things that happens as soon as a company goes public, then the public wants to take all of the profits and use it for whatever purposes they bought the stock in the first place. But the last thing they want is for the company to be doing research. They typically see this as a waste of money. And it really isn't so. It's very short-sighted. And I think if any of you are thinking of starting a company, you might consider doing it along the biolabs model. 75% of the staff are scientifically trained. And more importantly, we have an incredibly cooperative environment. Everybody at the company is friends. And a part of that is because the money that we don't spend on research or running the company ends up being shared among the employees at the end of the year. We're a profit sharing company. And really, it's made a huge difference to the ability of the employees to value what they do at New England Biolabs and to do everything we need them to do in order to keep the company profitable. Now let's talk about some of the things that have come out of New England Biolabs. In addition to restriction enzymes, we've made DNA polymerases, kinases, and really a lot of things, some of which have led to vaccines. So I'm sure during these last few years, you've known about the mRNA vaccines that have made a huge difference, at least in the West, not so much so in the developing world, unfortunately, but in the West, they've made a huge difference. And the mRNA vaccine, the one that is made by Moderna, uses 3 enzymes that we sell and that we found in the first place in order to be successful. These enzymes modify the cap structure and put on the cap structure onto the messenger RNAs. We also sell an enzyme that Pfizer uses in their vaccine. And so here we are in response to this worldwide crisis. And because the company had been set up in a very clever way, when we needed to scale up, and in fact, we scaled up some 10,000-fold in the production of these particular enzymes, Then we were successful, we were able to do it, and our growth was enormous. In fact, the company has quadrupled in size over the course of the last 2 years. Now, I want to turn away from New England Biolabs for the moment, because I think here you can see how very basic research in molecular biology has led to tremendous advances in biomedicine. Now let's go back to my time at Cold Spring Harbor. So after I arrived at Cold Spring Harbor and started working on restriction enzymes there, we began using some of these to map adenovirus DNA. And this led to a project in which we worked with Phil Sharp. In fact, Phil is the person I shared the Nobel Prize with, but it's the only paper we had together was a mapping of adenovirus DNA with restriction enzymes. But we were also interested in transcription, and in particular, I had a postdoc, Richard Gelinas, who came to me from Harvard. I'd met him when I was a postdoc myself at Harvard. And he came and worked with me. And we started a project to ask the simple question, do eukaryotic promoters look like prokaryotic promoters? That is, are they the same or are they different? And this was timely because no one knew what a eukaryotic promoter looked like at the time. This was in 1974. Whereas we did know what bacterial promoters, at least what the lac promoter and the promoter in lambda looked like. And so we decided that we were going to start in a very simple fashion. And the idea was, if you look at the transcription map on the right, you can see there are two messenger RNAs, E1A and E4, that are transcribed in from the very ends of the DNA. And we thought it would be good if we could find the 5' end of those messenger RNAs, and that was simply by looking for an oligonucleotide with a triphosphate on it, because that would be the starting point, and then sequence the DNA coming in from the ends, and then see where the promoter was, and then we would define the promoter. Well, we got started doing that, spent a lot of time trying to get enough RNA and trying to get the reagents we needed, but we were not very successful. And so then we switched to looking at late RNA, and the late RNAs are shown here, L1, L2, L3, L4, L5. These all map to different parts of the adenovirus genome, and they all overlap. They have different lengths and so on. And so we thought we would start with those because in this case, it was easily possible to get a large amount of RNA because late in infection, adenovirus stops making its early RNA and just makes late RNA. We were also very lucky in that several groups discovered the cap structure that was present at the ends of the RNA. And so what we did was to devise a method. I found a good method, or we made it up, in order to get the 5' ends because of the cap structure. So we devised a method that allowed us specifically to get the cap structure. And when we did that, we were expecting to see, just from the late RNAs, maybe a dozen or 15 different cap structures corresponding to where these RNAs were beginning. But we didn't get that result. What we got was just a single oligonucleotide that was found on all of these RNAs. Now, of course, the first thing that happens when you get a result like that is the experiment that you thought was going to be very revealing was a failure. And so we did a post-mortem to try to find out what the problem was. We did a lot of experiments over the course of the next couple of years, in fact, trying to convince ourselves, as well as others. Most people were skeptical. They didn't believe it. They thought we were looking at some artifact. But we were pretty certain that we were looking at something interesting. And so what we did was to discover a number of things. We isolated, and others isolated, 1 or 2 pure RNAs and showed that indeed they did all have this same structure. And we came to the conclusion that it could not be coded on the DNA right at the start of the message. That is, it somehow got onto the end of the message, either post-transcriptionally or by some novel transcription mechanism, in much the way that the poly-A at the 3' end of a message is added by poly-A polymerase. And so we thought to do an experiment that is basically described on the right-hand side here. So if you take a messenger RNA and hybridize it to the DNA where it is coded, and we knew where that was because of our mapping work, then the 3' poly A will stick out because it's not coded next to the 3' end of the RNA. And in our case, we figured the 5' end also would stick out, and perhaps there was a way in which we could find it. And I had a guess as to where that 5' end might be coded. And so we took a piece of adeno that we thought was coding it, made a suitable probe to try to form what was shown in the middle slide. So the green is the probe and the red is the RNA. And we figured that we would be able to see this probe DNA in the electron microscope. Now, the only problem with that was that neither Richard nor I were electron microscopists, but we were lucky because at Cold Spring Harbor, there were two excellent electron microscopists, Louise Chow and Tom Broker. They were married, and they worked just down the hall from us. And so we went down on a Saturday morning after I thought of this experiment and said, do you think you could do it? And they said, well, it's never really been done before, but we think we can. And so we made the reagents, or Richard made the reagents on the Sunday and the Monday, and then we took it down to Tom and Louise. And indeed, the first picture they looked at in the electron microscope looked rather like the picture shown in the middle, except that it was a little bit different. Instead of just hybridizing at one point to the DNA and to the messenger RNA, the probe DNA hybridized at two separate points, showing that what was going on was that the RNA was actually being encoded at two different points in the DNA and somehow was getting joined. Well, this was the discovery of RNA splicing, and of course that's had an enormous impact going forward. This was the paper describing that discovery. This, in fact, is the first time the word amazing was ever used in the title of a paper. And I have a small story here in that I call the editor, the referee said, you can't use it. It's not suitable to put the word amazing in a title of a paper. But then I called the editor, and during the course of our conversation, I said, well, what did you think when you first saw this? And he said, oh, I thought it was amazing. And I said, well, I rest my case. And so the word got into the title of the paper. So this was in 1977, sometime before the Nobel Prize was awarded in 1993. Now, as soon as we started working on adenovirus and trying to do all of this, one of the things we needed to do was to actually sequence both the messenger RNA and the adenovirus DNA. And we also were very interested in just what was being coded on the DNA. Where were the proteins? Where were the signals that were there? And so this is how I got into bioinformatics in the very early days. This was starting in 1977, actually, when we were doing this. And we wrote a number of programs that could help in assembling DNA, in analyzing it to find out where the proteins were, where the RNAs were, and also help to predict the dynamic properties of the organism. And of course, bioinformatics is now an absolutely essential part of biology. Now, one of the things that happened in parallel a little later from this was the Human Genome Project. And the idea of sequencing the human genome came up much later in 1985. Bob Sinsheimer and co. had a meeting and decided it was perhaps worth doing. DOE and NIH put some money into this. And by 1995, the very first bacterium, Haemophilus influenza, that Ham Smith had discovered Hindi2 and Hindi3 in, this was the very first bacterium to be sequenced, and Ham Smith was a part of that. Following year, the first simple eukaryote, yeast, was sequenced. And in 1998, the first complex eukaryote, C. elegans. Now, it was around this time that Celera Genomics announced that they were going to sequence the human genome, and I ended up being chairman of their scientific advisory board. The first thing they did, once they got going, was to complete the sequence, get the complete sequence of the fruit fly, quite a big undertaking. And then the first plant genome was sequenced, And finally, in 2001, Celera Genomics, as well as the publicly funded Human Genome Project, produced the first draft of the human genome sequence. In 2002, things started to go awry. The Human Genome Project tried to claim that Celera had failed, which wasn't true. But in 2001, the Human Genome Project in Nature published what they considered the complete draft sequence of the human genome. It was actually far from complete. And then we at Celera published in Science the complete sequence of the human genome. But it wasn't until 2004 that it was really close to completion. But again, there was still a lot of genes missing. There was a lot of DNA sequence that was not accounted for. And in fact, it was only earlier this year that anything approaching a complete sequence of the genome was ready. And that was, of course, a really huge impact, because that is going to make precision medicine a possibility. Oh, done it again. Let me tell you about one other thing that happened that relates to my work, and that was the discovery of base flipping. That is, when DNA methylases bind to DNA, they flip out the base so that it goes into a little pocket in the enzyme where the modification takes place, where in this case, methylation. And we now know that this mechanism of modifying with a variety of methods DNA bases is to flip them out, because they're essentially inaccessible when they're inside the double helix. We discovered this just before I had to go to Stockholm to pick up my Nobel Prize, and in fact made this the topic for my Nobel Prize lecture. But I don't want to talk about that. I want to talk about the fact that my recent work has all been on methylation in bacteria, and something very interesting has happened that also has medical consequences. In Clostridium difficile, this is C. diff, this is a bacterium that causes lots and lots of problems in hospitals. Patients frequently pick up these hospital-acquired infections, and C. difficile is a big problem. And the major reason that it's a problem is because it can form spores, that is, inert bodies that just sit there and wait for the antibiotic to go away or whatever other treatment, just wait for it to go away. When that goes away, then they start growing again. And so most C. difficiles have now become very resistant to antibiotics and very difficult to cure. In fact, fecal transplants are one of the few ways that they can be cured. But I was working on C. difficile, and I found an enzyme, a methylase in C. difficile, that recognizes C followed by 5 A's, and it methylates the last A residue. And it turns out if you knock out that methylase, C. difficile cannot sporulate anymore. This is controlling sporulation. And in the process, this means that if one could find a drug that would target it, then it should be possible to stop C. difficile and actually develop a cure for it, because it would stop the sporulation. Well, 2 clinical studies are underway. I've not heard the results yet, but I'm really looking forward to the possibility that this will be the very first drug targeting a DNA methylase. And the reason I'm excited about that is that there are many, many bacteria we've discovered that have methylases that are solitary, they're not part of restriction systems, and are likely controlling gene expression in some way or having fundamental impact on the ability of bacteria to grow. So I'm very excited about the future, and I hope that you've seen through this that by pursuing basic research and then allowing others to take it, doing some of it myself, that you can easily lead into biomedicine, into commercial opportunities. And I think basic research is something we've got to make sure we continue doing it. So I'll stop at that point, and I thank you very much for listening, and I'll be happy to take any questions. Thank you.
Thank you very much, sir. Is my voice audible? Thank you very much, sir. Is my voice audible?
Yes, I hear you.
Yeah. Thank you. Thank you so much for scintillating talk, Sir Rich. This session is chaired by Professor Deshpal Verma, Professor Emeritus at Ohio State University, who is here on site with us, and of course myself, along with Anil, who is a reporter. So the room is open for a quick Q&A. Sir Richie Roberts has a hard stop at 9:30. Which is a few minutes from now. So any questions, please? Yeah. So.
Richard is Deshpal Verma. I had a pleasure to meet you at Cold Spring Harbor in 1981, I think, yeah. So is the methylation process similar between the prokaryote and eukaryotes with respect to this cytosine methylation you mentioned?
Sorry, I couldn't really gather that. Could you repeat it, Prash, please? Because I can hear you more clearly.
So the question is the methylation mechanism is the same between prokaryotes and eukaryotic DNA?
Well, the enzymes are different. So the DNA methylase in eukaryote is actually quite a complex enzyme rather. It has a long N-terminal that actually allows it to interact with a lot of different substrates. And so in that case, it really doesn't behave in quite the same way. There are 3 well-known methyltransferases in eukaryotes, at least in humans, and they all behave a little differently. In bacteria, they're really all pretty much the same. There's not a lot of difference, but they just really look very much like the terminus of the eukaryotic methylase, and the eukaryotic methylase doesn't have the same specificity that bacterial methylases seem to have. We've only come across a couple of bacterial methylases, one that seems to methylate all adenine residues, and another one that seems to methylate not quite all, but a large number of cytosine residues.
Thank you.
Most of them are very specific in bacteria, not specific in eukaryotes.
Okay. Thank you, Richard. I'm Geeta, and I have loved your talk. I find it transformative that you chose not to be an author on a paper that is not open access. What is your message to young scientists across the world who need a career and therefore high impact papers, and yet they want to address the open access revolution? So we want to be part of the open access. What do you think is the future so that we can keep our careers intact, and yet I hope you understand my question?
Okay, so I feel rather strongly about open access, which is why I wouldn't put my name on any paper going into a journal that is not open access. We started a movement among Nobel Prize winners way back in about 1996 or 1997. We got a large number of laureates who all wrote to various funding agencies, to various journals, and said, consider moving to open access. Quite a lot of them have, but even today, some of them haven't. But finally, today, the NIH has said that within 2 years, all work that is funded by them must be open access. The British Research Council's got a little ahead of them. In Britain, everything that is funded has to be open access. And I think one of the things that we need to do is to make sure that the people, the bureaucrats in the universities who consider whether people should be promoted or not, are encouraged to actually read the papers rather than just look for artificial indices on impact factor and on other things. And in order for them to do that, they need to make sure everything is open access and that everybody can read it. I think it is not right that public funds should be used for research that cannot be read by the general public without paying again for it.
Thank you, Richard. That answers my question brilliantly.
Good.
Thank you so much. Sir Richard, I have a question. We have always been fascinated with the alternative splicing. And ever since this was discovered, a lot of emphasis has been in the rounds with respect to the differentially expressed genes and the gene expression studies. Is there any amount of seasonal plasticity that is attributed in lieu of alternative splicing, or the way we really look at the gene expression profiles these days come next generation sequencing?
Okay, could you just repeat the last part, because it wasn't terribly clear? Maybe Prash, you could.
Yeah, I'm sorry, I was the one who asked, Sir Rich. So what I was asking is there any amount of seasonal plasticity that is attributed to the alternative splicing events and commit next generation sequencing approaches? We have a lot of differentially expressed genes that are being upregulated or discovered. So is there any amount of seasonal plasticity with respect to the phenotypes that is attributed?
Okay, well, let me see if I can. I'm not sure still that I understood it, but if you're asking what is all of this extra DNA that's sitting in the introns between the exons that are spliced together, what is that doing? The bottom line is we really don't know. We're still trying to work it out. And in fact, if you look in the human genome, it's a very small percentage that we know is coding for protein. We know there is some that is coding for RNAs, and these RNAs have function, but often we don't know what they are. And there's a huge amount of information in the human genome that we simply do not understand. And I look upon this very favorably because I think it means that we biologists have jobs for a long, long time. We have to figure out what all of this extra information is doing.
Yeah, perfect. Thank you. Thank you so much. Yeah. We still have 3 more minutes. Yeah, please. Yeah.
Sir, I am Professor Krupanidhi from Vignan University, who wish to know through you, sir, about Combres project that we are handling. We have got good many number of bioinformatics students. They will get involved in that. Please elaborate on this Combres project.
Okay, I didn't get that at all.
Combres project that we are having, that is computational bridge experiments.
Combrex.
Yeah.
Yeah.
That our students from Vignan University, that is bioinformatics students, they wish to work with that. So what is the possibility?
Right. So we were very excited about Combrex when we first came up with it and we got it funded. And then it was funded for 2 years. We made tremendous progress. And then NIH, in their wisdom, decided they weren't going to fund it anymore. And they stopped our funding. And so I was incredibly disappointed because one of the things that came out of that project that made very clear is that we really need to establish the function for as many bacterial and human and other genes as we can, and to do it in a way where we understand the biochemistry and we understand the genetics. Combrex was really aimed at the biochemistry, and we had a lot of success. But one of the things we realized was that there was no gold standard database of proteins. So if you go and look and you ask, well, what proteins do we really know the function of because they've been characterized experimentally? It doesn't exist. There is no gold standard database out there. And so an awful lot of what we think is function is just computer predictions, which may or may not be accurate. And so we need to find a much better way of categorizing proteins, of knowing how far it. So I take a new protein, I get the sequence, I look and see what it is similar to. We need to know if the thing that is similar to has been biochemically characterized or genetically characterized in some way. And so I'm hoping that some other groups will pick up on the Combrex project, but unfortunately we don't have the funding to do it anymore. So it really is very sad.
So our students wish to do the work online in this Combrex project, particularly related to phase display, short chain fragment variable things.
Sorry, the sound is terrible from your end. Can you maybe say it again, but slowly?
See, short chain fragment variable. Short chain fragment variable. that is being displayed by the face. So that has become a short protein out of immunoglobulin, which is having 150 kilodaltons. So this is only 25 daltons, kilodaltons, 25,000.
Yeah, I apologize. I really can't hear that well enough to provide a clear answer to anything.
Very good. Thank you, sir.
I'm sorry. If you want an answer, I suggest you email me and ask the question, and I will respond by email. My email address is roberts, R-O-B-E-R-T-S, at NEB for NewEnglandBiolabs.com. So just send me questions. I always answer them. I always answer my email.
Thank you. Thank you so much, Sir Rich. We could not have expected wonderful beginning without this scintillating talk by Sir Richie Roberts. Friends, it is absolutely a wonderful take-home message here. Sir Richie Roberts.
Thank you.
Before we close, maybe have a quick round of applause and give a standing ovation to Sir Richie Roberts, please, for the yeoman's service that he has been doing over the years. Thank you so much. Thank you. Thank you so much, sir. We're absolutely elated, and thank you so much for obliging to our request amid being your busy schedules. Thank you. I'll just take opportunity in thanking Sir Richie Roberts and, of course, Professor Kavikishore, sir, whom we have been trying. The last paper that Sir Richie Roberts cited, we had an immense opportunity to cite that particular paper. And when we are ending the Open Access Week today, I think this is a perfect tribute by Sir Richie Roberts to Open Access community, where he declined to be an author in his own works. That's in fact a kind of a great tribute that Open Access does. Thank you so much, sir. Thank you, Professor Krupanidhi, for introduction. Yeah, we'll just move to the next phase.
Thank you, Dr. Prashant, sir, for compiling the gist of the program. And as sir rightly said, it is indeed a great privilege to have this wonderful beginning for the 3-day international conference. And it's indeed wonderful to have heard so many interesting pages from the sagacious journey of Sir Richards. And then I think amazing is the only word we all can use, as sir said. That's a word he put in his paper. So we should all acknowledge that amazing is the right word for the kind of work sir has done and for the kind of lecture sir has given. Can we have one more round of applause for Sir Richard and also for the organizers for bringing him online for all of us. Thank you. We will shortly begin the inaugural session for this conference. We are awaiting the arrival of Professor P. Nagabhushan, sir, Vice Chancellor of Vignan Groups. So once he arrives into the hall, we will start the inaugural session. Thank you. The formal inaugural session was scheduled at 9:45 a.m. Since we finished the first session a little early, we have this buffer time of 10 minutes. So we will wait for the arrival of our Vice Chancellor, and then we will begin the inaugural session. And this is a kind request to all the students. Please be seated. Do not roam around. In 10 minutes, we will be beginning the formal inaugural session. Thank you. Have this buffer time of 10 minutes. ఒక్కసారి సార్కి చెప్పండి. వైస్ ఛాన్సలర్ ఉండని సార్ ఉండని కాదు, ఆయన ఎంపీ గురించి ఆగండి అంటున్నారు, కవికిషోర్ సార్ వద్దంటున్నారు. ఏం చేయాలి చెప్పండి నాకు ఒకసారి క్లియర్గా స్టార్ట్ చేయండి. I once again extend a gracious welcome to you all for this international conference on frontiers on nutrition, medical genomics and drug discovery, INBIX'22, being organized by Department of Biotechnology and Bioinformatics, Vignan groups under the aegis of bioclues.org. A famous Swiss psychologist, John William, once said, "Scientific knowledge is a perpetual evolution. It finds itself changed from one day to the next." In this wonderful journey of science between an old today and a new tomorrow, international conferences like this provide us a wonderful platform to explore science in its true sense. To have so many stalwarts of science from across the globe assembled here, it indeed is a great honor for all science lovers like us to be a part of this wonderful conference. So let us now begin this event formally by having our dignitaries on the dais. May I request Professor P. Nagabhushan sir, Vice Chancellor of Vignan Groups, to please join us on the dais to welcome all the other dignitaries. Professor P. Nagabhushan sir, former director of IIIT Allahabad and having held various responsible positions. Today he is working hard to make this Vignan Groups a significant and a magnanimous university. Thank you, sir. As I already mentioned, it's a great honor for all of us to have Padma Bhushan Sri Professor G. Padmanabhan sir as the chief guest of this magnanimous event. Can we put our big round of applause to welcome the chief guest of today's conference, Padma Bhushan Professor G. Padmanabhan sir, former director, Indian Institute of Science, Bangalore, and chair, BIRAC. Sir has played an integral part in the growth of life sciences and biotechnology in the country. Having mentored students for over generations, Sir is a prolific and decorated researcher. Sir, it's a great honor for us to have you amidst us today. I would now like to invite our guests of honor for today's program, Professor D.P.S. Varma sir. Sir, we request you to please join us on the dais. Sir is FRSC from Ohio State University, a world-renowned cell and molecular biologist with over 200 research publications, many books. He's an international speaker, and for the last 40 years, he's been traveling across the globe talking about science. Sir, it's indeed an honor for us to have you here with us. Our guest of honor, Sri Lau Sri Krishna Devarailu Garu, MP, Lok Sabha and Vice Chairman of Vignan Groups will be joining us shortly. I now request Professor T.C. Venkateshwarlu sir, Head Department of Biotechnology and Bioinformatics, Vignan Groups, to join us on the dais. May I now request Dr. Anil Kumar, Organizing Secretary of INBIX'22, to also join us on the dais. I once again thank all our dignitaries for being with us today. Sir, may I request all of you to please mark a ceremonious beginning by lighting the lamp. we are custodians of a very rich tradition of heritage and culture, and we strongly believe that this lamp that is lit today ignites our minds and soul and builds in that scientific temper in all of us. I thank all the dignitaries for making an auspicious beginning. May I now welcome the students of Vignan University to render the invocation on Vignan theme.
Check, check, check.
जय जय विद्या जनयित्री, जय जय विज्ञान साम्राज्य। जय जय विद्या जनयित्री, जय जय विज्ञान साम्राज्य। जय जय विजय संदात्री, जय जय यावत् प्रपंच। विद्या विज्ञान महिमान्वित, जय जय विज्ञान महिमान्वित। जय जय विज्ञान महिमान्वित। विद्या विज्ञान महिमान्वित, जय जय विज्ञान महिमान्वित। विद्या विज्ञान महिमान्वित, जय जय विज्ञान महिमान्वित। अक्षरमणिमय माला परिशोभित शुभगात्री, अक्षय विज्ञान किरणा भासुर संपद्धात्री। विद्या विज्ञान महिमान्वित, जय जय विज्ञान महिमान्वित। विद्या विज्ञान महिमान्वित, जय जय विज्ञान महिमान्वित। सांकेतिक सद्विद्या संकलित ज्ञान नेत्री, सदाचार सद्बुद्धि प्रदपतित संदात्री। विद्या विज्ञान महिमान्वित, जय जय विज्ञान महिमान्वित। विद्या विज्ञान महिमान्वित, जय जय विज्ञान महिमान्वित। विज्ञानाम्बर विहारा विमलद्युतिकारयित्री, प्रज्ञान प्राभव सन्धानित सौन्दर्यधात्री। अज्ञानध्वान्तहर सुधाकर शारदरात्री, सुज्ञेयप्रद विलासा सुन्दरतर भावयित्री।
I thank the students for rendering such a wonderful song which talks about the aim of true education. Thank you. It's an honor for all of us to have with us the Vice Chairman of Vignan Groups, Honorable Lau Sri Krishnadevarailu Sir, MP Lok Sabha. Sir, thank you for joining us. May I now request Professor T.C. Venkateshwarlu Sir, Head Department of Biotechnology and Bioinformatics, to deliver his welcome address.
So a very good morning to the chief guest, Professor G. Padmanabhan Sir, Padma Bhushan Awardee and former Director IISc Bangalore, and guest of honor, Professor Deshpal Verma Sir from Ohio State University, USA, and beloved Vice Chairman of Vignan Group of Institutions, Dr. Lau Krishnadevarailu Sir, Member of Parliament Lok Sabha, and our Honorable Vice Chancellor Sir, Professor P. Nagabhushan, Vice Chancellor of VFSTR, and my dear students, faculty from Department of Biotechnology and Bioinformatics. I am happy to welcome you all to this international conference on frontiers in nutrition, medical genomics, and drug discovery. So the morning session started with a eminent and excellent talk by Dr. Sir Richard John Roberts, a Nobel Laureate. So Sir Roberts, along with Philip Sharp, successfully demonstrated how mRNA can be divided up into introns and exons, so which is the discovery of RNA splicing. So on listening such a great personality, the young students with the tender minds and faculty of VFSTR are really motivated. So the INBIX'22 conference is going to cover the various aspects relating to the trends and innovations in proteomics, genomics, and molecular medicine. So this conference helps the students, scholars, and faculty of VFSTR and also faculty from other institutions in understanding and execution of research relating to the molecular medicine and drug design. So this conference provides a platform and valuable recognition for the speakers and poster presenters to present and discuss the various trends and practical challenges so that are encountered and solutions adopted. So with this, we look forward to learn the latest updates on current aspects of medical genomics and drug design from eminent speakers. So we look forward to have a collaboration with all experts present over here. So the faculty of biotechnology and bioinformatics are working in areas like genomics, immunoinformatics, in silico vaccine design, and drug design. I request all the experts to extend your warm support in research, so this could benefit our students, including faculty, in achieving various scientific goals. Thank you. Thank you, sir.
Thank you, sir. Thank you for making such magnanimous efforts in making this conference possible. Thank you. May I now request Dr. Anil Kumar, Organizing Secretary of INBIX, to introduce all our dignitaries on the dais to us.
Thank you, madam. Welcome to the dignitaries on the dais and off the dais. My dear colleagues, scholars, and students, I extend a warm welcome to all of you. I am privileged in introducing one of the keynote speakers, Professor D.P.S. Verma from Ohio State University. I'll give brief biodata of D.P.S. Verma, sir. Professor Verma is a world-renowned cell and molecular biologist at the Department of Molecular Genetics, Ohio State University, Columbus, USA, where he worked for over 33 years and contributed to the overall development of several interdisciplinary graduate programs, including molecular, cellular, and developmental biology. He is the one who has isolated mRNA for the first time, and he is a doyen in the field of biotechnology. He has received several awards in his career. I would like to mention a few of them. His early career started at the McGill University, Canada, where he received National Research Council State Award by the Governor Canada for being the best young scientist in his field. He was also received several other awards like Third World Academy of Sciences, Italy. He received Doctorate Honoris Causa for first plant mRNA isolations at the University of DPK, France. Professor Verma has pioneered in the field of biotechnology, starting from the isolation of plant mRNA, cloning first gene discovery of many genes such as nodulins, which are being widely used in industrial purposes. His work led to the creation of several companies in Canada, which include Monsanto, DuPont, Bayer, and Matrix Labs. He is an advisor to the Planning Commission of Indian Agriculture, where he has submitted 300-page report, How to Improve Agriculture in India, a report by name Taxa report, which you can get from Google. I am honored in introducing our guest of honor, Professor G. Padmanabhan, Padma Bhushan Awardee, Senior Science and Innovation Advisor, BIRAC, India. Professor G. Padmanabhan, born in the year 1988, and is the first student of Indian Science Institute of Bangalore to become its director. He contributed significantly to the initiation of recombinant DNA research in the country. He had initiated on liver cytochrome P450 gene expressions, which plays a crucial role in drug metabolism. The molecule heme has been at the center of his quest to understand its multifarious roles. He has been working with the malaria parasite biology for the parasite development during the transmission stages and parasite virulence in its asexual stage of development. This pathway, includes a drug target against several malaria. He has also shown that curcumin from turmeric has tremendous potential to treat malarial diseases. He has produced over 50 PhDs and published over 180 papers. His letter in Science explaining the background behind India for Pokhran nuclear test was widely appreciated. He has written his memoirs on his experience in doing science in India in 2008 and 18. Professor G. Padmanabhan was a visiting scientist professor at the State of Chicago for a decade, since 1973 to 1986. He served as an organizer for several international organizations such as WHO, ICGEB as an expert member. His profile was covered in Nature Medicine. He continues to be active in research after his formal retirement in 1998. He has mentored a generation of students, juniors, and senior scientists, and later they become entrepreneurs. He chairs several technical committees for the Government of Science and Technology establishment. He was elected as President of National Academy of Sciences, NASI, in 2009. He has been the Chancellor of Central University of Tamil Nadu since 2018. He is serving as a Senior Science Innovation Advisor of Biotechnology Research Assistance Council, BIRAC, of DBT, Government of India. He was awarded the UNESCO Chair in the year 2000. He was co-chair for the COVID Vaccine Evaluation Committee of DBT. Now I would like to introduce our Vice Chairman, Sri Lau Sri Krishna Devarayalu. Lau Sri Krishna Devarayalu, born in the year 1983. Right now he is serving as Member of Parliament, Lok Sabha from Narsaraopet, and is also member of Standing Committee, Minister of Human Resource Development. In addition to this, he is serving as Vice Chairman, Vignan Groups. Since last 4.5 to 5 years, he is doing human services for promoting high education standards in India and contributing the socio-economic development in this particular region. Thank you one and all.
Thank you, Dr. Anil sir. It's indeed a great excitement for all of us to listen about these scientists who've been dedicating years of their life to make science possible. Thank you. May I now request Dr. N. Jalaja, convener of the conference, to explain us about the conference INBIX'22.
Namaste everyone. I myself, Dr. Jalaja, Associate Professor, Department of Biotechnology. As a convener of INBIX'22, I take immense pride in organizing this international conference on frontiers in nutrition, medical genomics, and drug discovery of our lush, greeny Vignan University. On behalf of the organizing committee, it is such a rarity that a Nobel laureate and the several other galaxy of scientists have come all the way to Guntur and elaborating the latest developments of bioinformatics and medical genomics. After COVID, it has been the first time we all are meeting on site and Vignan University to get this opportunity to organize INBIX'22 under the aegis of BioClues is the immense pride. Our chairman, Dr. Laureate Garu, and vice chairman, Mr. Sri Krishna Devarayal Garu, our vice chancellor, Dr. P. Nagabhushanam sir, and registrar, Commodore M.S. Raghunathan, and Dean School of Biotechnology and Pharmaceutical Sciences, Professor S. Krupanidhi, and HOD Biotech and Pharmacy have been very helpful in organizing the conference. We received a total of approximately 350 abstracts, over 700 participants registered for this conference, which is biggest so far. Conference, we hope the young and old take part in this August conference and deliberate excellent science in making INBIX'22 a grand success. We thank all the speakers, participants in attending this conference, and also students. Come, let's begin INBIX. Thank you.
Thank you, Dr. Jalaja. We appreciate all the meticulous efforts put forward by the entire organizing team to make a conference of this magnitude possible. Thank you. May I now request Professor J Raman sir, Flame University, Pune, Advisor-in-Chief, bioclues.org, to talk to us about BioClues.
Good morning, all. At BioClues has entered 18th year of existence. As an advisor, I take pride in BioClues and her achievements. At BioClues, we wish to create and disseminate consciousness of research in the field of bioinformatics and system biology through webinars. We wish to computational biologists. We wish to impart a complete understanding of the principle of bioinformatic algorithms and tools through interactive summaries, polls, and discussions. We wish to provide an online project platform for mentoring graduate students while bridging the interaction of students in the field of computational biology. We want to cater to the needs of the assimilating existing open source code and developing novel tools and web servers for research community. We wish to encourage women scientists take up bioinformatics. BioClues, as an international bioinformatics academy, is bridging the gap between Indian researchers and international researchers in a single forum. Women bioinformaticians are more empowered than now, and Indian bioinformatics has a real balanced gender ratio. All the bioinformaticians of India are under the umbrella of BioClues, and we are making the path of multidisciplinary research and making a proper mentor-mentee relationship for bridging the future generation in the same boat. As BioClues already started the Bioinformatics for School Children initiative, maybe in the year 2030, we will have motivated and trained bioinformaticians leading India in big data analysis. We have grown tremendously thanks to the leadership of Professor G. Nahari Sastry as President, Sugunakar Vuri and Harpreet Singh as Secretaries during the last time. Currently, on behalf of our founder, Prashant, and BioClues, we welcome Jnaneshwar Chaube as the new President and K. Sri Manjari as Secretary. I am sure the new team will bring leaps and bounds of activities with joy. I wish you all take the conference to greater heights as envisioned by able leaders. INBIX has been taking leaps and bounds with successes. We thank the organizers at Vignan University for this stupendous effort at INBIX'22. Wishing you all success in the conference.
Thank you, sir. Thank you for taking up this stupendous responsibility of making multidisciplinary research possible through BioClues. Thank you. It's time for all of us to listen from our guest of honor, Professor Deshpal Verma, sir, FRS Biotechnology Center, Ohio State University. Sir, we are eagerly waiting to listen from you.
Good morning to the young scientist, emerging scientist in the country. I have only one message, and that message is learn to imagine things and then make it happen, because it's all within your power. This feeling of helplessness that I can't do this, I can't do that because of this, because of that, you got to get out of that framework of mind and then move towards achieving new things. And new things can only be achieved by having an internal passion, because nobody can make you achieve anything, nothing. Parents can't do it, teachers can't do it, society at large cannot do it. Just take some inspiration from me, please. I come from a place where there was no road, no electricity, no water. The fortunate thing was that there was a school there. And that school was built by a philanthropic person who went back to the village and built this school, high school. But that school stands even today, and that was probably one of the best schools in the country at that time. But being curious from the childhood, I learned so much within high school that even today, I remember everything which even the PhD students of today don't know. The first question I ask to most of the guys in the lab or associating with the companies, can you boil the water at 0 degree? And they have no clue. These are graduates of today. They have no clue that you can boil the water at 0 degree. Why? Because they are not applying what they learn, that you can indeed boil the water at 0 degree if you change the pressure. So this basic thing that you learn in high school, you got to use that basic knowledge, and if you haven't learned, then go and relearn it, because that is the fundamental thing which is going to let you move forward. Publishing papers and collecting data and all that analysis is very good just to get more salary, promotions, but you cannot innovate things. Innovation comes from your heart, from your passion, and trying to accomplish things that you want. So, coming from that kind of a background, the only thing I had was curiosity. And that curiosity took me all the way where I am today. I have helped start a number of companies, even in India. Avasthajan was started with my help. Max Lab in Hyderabad was started with my help. So there are LLX that in Toronto, that was started with my help. Agrigenetics in California, that was started with my help. So I have been involved in a number of platforms and had an opportunity about now 12 years ago or something like that, where I looked at what's going on in India with respect to the application of science to agriculture. And I gave a talk at the Planning Commission when Ahluwalia was there, and he said, stop, let's do something. He didn't let me finish the talk, and he immediately constituted a committee, gave me the charge, and I put a team of people together to look at Indian situation. Being a molecular geneticist, I really had no idea of the agriculture except what I had learned as a child in the village because I grew up in a farm family, so I have plenty of idea. Actually, I worked in a sugarcane field with these hands, so I had a pretty good idea actually what agriculture was all about. And it has not really changed much with respect to the farmer in India. So what I did, I really looked at all over India. What is the biosphere is telling you? What is the resource available? I traveled practically to all the institutes from Palampur to Trivandrum to Odisha. I even went to tribal areas to see what they can do or what they are doing, how we can help them. Do you know there are 10 million tribal people in this country? 10 million. Just go and visit one of the tribes and your eyes will open up completely how they are doing and what they can do. They have a lot of biotechnology potential buried right there in their backyard. So applying all this knowledge that you accumulate to the practical purpose, you can simply download this report. Is on the website which I built, secondaryagriculture.com. I coined this term, secondary agriculture. So you can look at this Taxa report, which is about 300-page report, looking at all bioresource that India has and what can be done using that bioresource. Now, applying the advanced knowledge that you're accumulating through bioinformatics, the purpose of bioinformatics is not just to collect the data and publish a paper. You have to collect a meaning out of this, what it is that this data is telling you and how you can use this data to apply to the real world problem. And in the process, you can make your millions and billions what you want to do, which you cannot do any other way. And Sir Richard was a clear example of doing basic research and how he became billionaire himself. He's a billionaire man because the company is highly valuable. I think it's right now more than $5 billion company. So my message to all of you young people is really start dreaming, and you can fill your dreams if you apply your energy in the right direction. Thank you.
Thank you, sir. Thank you for such an inspiring message. And the way you have transcended all the physical barriers to get yourself educated, and today you're an inspiration to millions of science learners. So let us all take this message home, that being curious and passionate is the only way to do science. Thank you, sir. May I now request our guest of honor, Sri Lau Sri Krishna Devarayal Garu, Vice Chairman, Vignan Groups, to address all of us.
Good morning, everyone, and a very warm welcome to this international conference. I start off with Sir Richard Roberts, who has taken his time off. I heard he was hit with COVID. Even then, he was supposed to be here, but because of some ill health, he couldn't be present with us. But he took his time off and made himself available, at least online, and addressed all the students and professors and science graduates here. And also a warm welcome to Professor G. Padmanabhan Garu, who's sitting with us on the dais, and DPS Verma Garu, who just tried to give his side of the story and inspired a lot of science students here. And Professor J Raman Garu, who just had an opportunity to speak, and TC Venkateswara Garu, HOD, and most importantly, P. Bhushan Garu, our Vice Chancellor, and all the participants who have traveled long distances to be here to participate in this conference, and more importantly, the students who are very enthusiastically for the last, I know I have seen them for the last couple of months, they put everything together and make it happen. So a warm welcome to all of them. And to start off with, the title of the seminar says Nutrition, Medical Genomics and Drug Discovery. I'm not a biotechnologist or I'm not deeply into this science, but what I could understand as a layman, looking at these three topics on the as a main as a title of this conference, I don't think nutrition, medical genomics, and drug discovery. More than this, I don't think anything you can fit in to actually make something out of it in this country. These three things put together, I think nutrition, as a layman, I can tell you, from the country which has the highest diabetic patients, and also one of the youngest population as of now, but next 30 years, we can see where it is going and how much healthcare needs to be evolved in the direction. And also with respect to the medical genomics, as a country which has one of the highest exports in terms of the service industry, either in IT or allied services, I think we are very well placed with respect to exploring this medical genomics, whether as Varma Garu just said about bioinformatics, how not just extracting the data, but actually how you can make the data much more relevant. I think we have enough human resources available, and we are doing it in the IT sector. I'm sure we can transcend that into this medical genomics also, make it much more relevant, as Varma Garu just said, and also make it much more affordable to all the citizens of the world. We have done that in multiple sectors. I'm sure we should be able to do that in the medical genomics also. And coming to the last one with respect to the drug discovery, most of you here, I don't think it's more than a month and a half. There was one gentleman who have come here, Krishnala Garu. He was with us and he was able to spend quality time with us. I don't think we need any better example than him. With the interactions that I had with him, we might be thinking all the research, all the new drug discoveries that only happen in the West, but here is the example of someone who could sequence the virus much faster or on par with any other Western country, and able to find a new drug to battle the COVID virus. So all of this put together, I think you are in the right place. Right place in the sense, not just the university, I'm saying right place in this country at the right time to actually explore these three things. I hope you take whatever Varma Garu just said about don't just read through it or try to apply the science as much as possible. I'm sure as Richard Roberts has done with his company, as he was mentioning earlier, how he could be able to raise such a company to such a valuation. I'm sure coming from, even from a rural setting like this, I'm sure there will be one or two people who will be looking in the direction, because this is the century of India. I'm sure you will take these opportunities with you and make it big for this country. Once again, for all the participants here, a warm welcome. I'm sure we made all the arrangements to the best possible. I'm sure there'll be one or two hiccups here and there. Please bear with us, but we made sure that this conference goes in a bigger and better way, and I hope you will take something, not just the researchers here, but the students who come from across the country. I'm sure you will take something out of this and come up with something new new in this country. Thank you very much.
Thank you for your message, sir. As one of the youngest parliamentarians in the country, you all will always stand tall in our vision. Thank you for being a backbone for this conference. Thank you, sir. May I now request Dr. Ratnagiri Polavarupu Garu, President and CEO, Genomics Carl USA India, to join us on the dais for the release of turmeric tablets, chocolates, and turmeric masks that were prepared in collaboration with Vignan groups. May I request our chief guest, Bhushan Sri Padmanabhan sir, to please join with Ratnagiri sir in releasing this. Can we put our hands together for this new discovery? A small attempt in making science come a long way.
Good morning, everyone. Everybody may be wondering what it is. And there are three important things in this, the small pocket, and it is nothing but curcumin. Why curcumin is so important and why we are releasing, in what form you are releasing. This area is full of curcumin around us. But the whole world is looking for curcumin for, importantly, to fight for the COVID. Professor Padmanabhan, Professor GP, very respectfully we call him, has given his life working with curcumin, and he's having a very tremendous knowledge about this and also how it works. And he is the backbone inspiring me day in, day out, and making these products, translational research, from the product, from the bench to bedside, how to make the curcumin into real product. One is chocolate. Why we are using chocolate? Because everybody loves chocolate. All the kids love chocolate. But if I put curcumin, normal chocolate 5 rupees, 10 rupees, but the chocolate is more medicinally valuable, which can help you to fight with viruses, bacteria, inflammation, and it is a great product which our generations we have been using. Same thing, mask. Normal mask is okay, but this is impregnated with curcumin, which fights, even stops the virus, especially COVID kind of thing, will stop there itself before it goes inside. And then we also have tea, curcumin tea we are going to use. I think today we are going to see it. And those are the three things we have: chocolate and a mask and also tablets, curcumin tablets. So we have them, and we brought almost like 100,000, and we are going to give some. And I think Krishna Devarayal Garu, in the lockdown time, he distributed throughout our area. I think he is remembering all those things, and he used to call us and, yeah. Yeah. All right. Thank you for inaugurating them. And I really thanked Professor Padmanabhan Garu to give me inspiration to make these things, how to make from bench to bedside, how to also we worked on these things, how it fights for the COVID. So please use curcumin, use healthily and usefully, and it is very, very good product. Thank you. It is from our area. We are from this area, so thank you.
Thank you. I thank all the dignitaries, and we congratulate this innovative idea. And I think now the tea break has become more important for more than one reason. We are also eager to taste those chocolates. Thank you. It is now time for us to listen from our chief guest, Padma Bhushan Professor G. Padmanabhan sir. Sir, may I please request you to address us?
Thank you. I have a technical talk to give later, but just now I'll confine to the curcumin story. You know, turmeric is known since Vedic times. Curcumin was isolated in 1825. If you look at Charaka Samhita, you know, those treatises, valuable treatises, there is no disease which has not been treated by turmeric or curcumin. I'm not exaggerating. From asthma to wound healing to whatever, that has been used. And therefore, I should congratulate Giri Garu. I don't say all that. You know, and he just puts my name. It is this, yes, I should compliment him. He says, I come from turmeric growing area. And yesterday we were supposed to go, but anyway, tomorrow he said he will take me to turmeric fields. But it is all his idea, everything. Of course, he will call me and then I am a sounding board, basically. It's the original idea, it belongs to him. And I think, I should caution you. Western world uses it extensively, especially for autoimmune diseases, arthritis in particular. I get emails. But suddenly you will see a group saying, oh, turmeric causes the liver problem. How many? 5 cases in 5 years. I mean, that they make a big deal out of it. So, and our people immediately will say, so and so abroad say this. Are they wiser than us? Millions of years we have been using this one. I'm not saying don't overuse, exaggerate, but many things have been done after a lot of thinking, and it is one product which is extremely useful. And I should think, and our forefathers knew what it has. Perhaps they didn't work out the mechanism. I thought I should at least take it up all the way in one disease and prove it. I took it for malaria. And I've gone up to the clinical trial, and we are expecting National Institute of Malaria Research to do the trial. I'm not an MD. Only MDs can do the trial. So we are hopefully, COVID came in between. Hopefully the trial will be done. So I don't want to talk about this conference. Maybe I'll say in my technical talk. This time I will just confine myself to the curcumin story. It is something we need to be. The region, I am told, is a major exporter for the whole country. Now, this is something we all need to be proud of. I don't know whether the students know you are in a turmeric belt. What should I say? No, you are right there. So it is something. And turmeric has more than 250 compounds. Chinese have analyzed it, not we. And curcumin is only one major component. There are many other volatiles, other compounds, terpenes, so many compounds are there. It's worth looking at their mechanisms. That is very, very important. It will add weight, value for the product, And you have to publish it. Publish it in big international journal. This is what it does. So that, few fellows who come in between, this is, causes a liver problem. I mean, I don't know. Look at how toxic are cancer drugs, and turmeric is a food product. We consume it all the time. So that's where I will stop. Wish you all the very best, Gary, and it's a great idea of yours, and all the very best. Thank you.
Thank you, sir. Thank you for always nurturing such small inventions. And we are really curious to listen to your session after this. Having a celebrated scientist like Sir Amit sir talks about the success of the conference. Thank you, sir. May I request all the dignitaries on the dais to please release the book of abstracts. I thank all the dignitaries for releasing the abstract book. I now request our Vice Chancellor, Professor B. Nagabhushan sir, to give his presidential remarks.
Good morning to all of you. You are all young scientists and you don't have a response at all. Good morning to all of you. You are very young scientists, very, very calibrated, very active, should be very active all the time. So the theme of the conference is quite interesting, has been reproduced by our Lavu Krishnadevarayalu also. But I look at it from a different angle. It tells frontiers in nutrition, good, medical genomics, very good, and drug discovery, excellent. Obviously, as Krishnadevarayalu has already mentioned, I am also totally novice to this area. My area is computing, my area is electronics, so I cannot speak heavily on that. But all that I could see is that most of the thing it is focusing on the physical health of the person. I am more worried about the mental health. I am more worried about not being egoistic. Ego is the greatest mental health problem, and I don't think all these things would cure this problem. The only solution, only cure to this problem is what Professor Verma just now from Ohio State University mentioned, that you start dreaming and you start working, as Professor Padmanabhan just now added, for the welfare of the society, for the entire health of the society, which surely assures a greater mental health. I'm very sure the youngsters sitting over here, the young budding scientists sitting over here, have got to think about this component more seriously and more rigorously. His call that people should start dreaming has really been excellent. He also made a very interesting statement that publications happen. After all, publications should happen. If you do something, it has got to happen, because I still know one basic statement, that for every paper there is a journal, and for every journal there is a paper. So for every paper there shall be a journal, and for every journal there has got to be a paper. That is not important. What is really important is the total social welfare that happens because of science, because of research. I'm very sure the youngsters, the young budding scientists sitting over here should draw the clue from all the elders over here that the science, the research is not for a stupid publication, and a stupid publication is not for the award of a PhD degree, and a stupid PhD degree does not really mean a big research. Research is the one which takes not only India, the entire globe to an excellent frontier end. I have got to congratulate here Professor Kavi Kishore, who has been the person behind the screen and who could get this entire program totally steered in a very excellent direction. I have got to recollect the role played by Professor Kavi Kishore. I'm sure he's standing somewhere. Okay, he's standing somewhere. Vignan, even when I was not the Vice Chancellor of this university, when I was just invited over here for some time ago, I got to mention, I went through the entire campus, sure. And without mincing words, I have got to mention that for whatever the reason, the biotechnology department really attracted my attention. I was very sure biotechnology department should turn out to be the jewel of this Vignan campus. I am very hopeful that it shall remain to that glory. I am very, very positive about that. Well, the other point which I just wanted to share with all of you is that the work, the research, whatever, as just now mentioned by Professor Verma, should not be just for the sake of a publication. After all, once you conceive, naturally delivery has got to happen. But then, if delivery itself becomes more important, people would definitely lose the importance of conceiving the idea altogether. I'm very sure when you hear these prominent people who have made science possible, you would really be motivated that what drove them crazy into science is the requirement that they have got to come out with something which excels. I'm very positive all the youngsters who are not only from Vignan, who have come over here to make the presentation in this campus, will surely be lured into this prolific area of excellent science and excellent science for excellence. And the excellence comes from a devoted mind, and excellence comes because of a patient mind, and the excellence is because of the mind which is capable of accepting everything, not merely the success, but also the defeat, because the defeats are plenty, the failures are plenty before one success could be realized. I'm very sure these success stories would be shared. I'm very sure you would be interacting with all the elders over here who would definitely tell you that the success was the last pedestal, but the steps up to that were all the steps taken second time, third time, and the fourth time. And those steps are very, very necessary. I'm very happy that this is a 3-day festival of learning, and learning is more important. But unfortunately, our whole entire education system in India is so bad and so collapsing that whenever education system from any university just aims at getting a mediocre job for a candidate in the name of a placement, where can the original thinking become possible? The original thinking has got to become possible, provided we induct that original feeling of thinking, original experience of thinking, right from the school days. And from the school days, this is totally missing because our mothers particularly and our parents totally are responsible in ruining the entire primary education system. So also our education system in India, which does not challenge any learner to learn, because all learners are not learning for examination. All these learners are the mediocre people who get into the old question papers to secure a lot of marks, and that becomes the end of learning, and with this becoming the target of learning. And finally, when they come into a bachelor's degree, their target is only a common, ordinary placement, and if that becomes the target, where can the science happen? Where can the research happen? Where can the creative thinking become possible? I'm very sure the youngsters over here should not lose the thread of getting into creativity in thinking, getting into critical thinking. And once that can be possible, the real science can be possible, and it can be possible in India also. Right? Why it should not be possible? Well then, when people address me as Vice Chancellor, somewhere I feel that I have lost the clue of total research. I would love to be addressed as lifetime professor at IIIT Allahabad, because that clearly tells that I am still into research. Vice Chancellor unfortunately has got to happen because somebody has got to take the responsibility of leading the institution. And this leader becomes more important when the ship starts sinking, because somebody has got to be there to blame for the sinking of the ship. And that blame to when the ship starts sinking will be taken up by the vice chancellor. But then the lifetime professor in vice chancellor has got to instigate every one of you that originality lies in critical thinking, originality lies in creative thinking, and originality does not lie in reproducing the answer in a stupid examination or in a stupid test system. The creativity in every question which is asked by a teacher could prompt the birth of a scientist, could prompt the birth of a researcher, could prompt the birth of a thinker. I'm very sure this conference would give rise to many, many thinkers here. And there are plenty of thinkers, but the thinkers have got to bud out. Thank you very much for such a patient listening.
Thank you, sir. Thank you for that wonderful message and a well-summated address. And as sir lightly thrown light on the fact that a healthy mind is very important for happy learning and creative thinking. Thank you, sir. It is time for us to felicitate our dignitaries for the precious time that they have given us and also for the valuable words that they have shared. May I request Vice Chairman Sri Krishna Devaraylu Garu to felicitate our chief guest, Professor Padmanabhan, sir. I request our Vice Chancellor also to join in felicitating our chief guest, Padma Bhushan Sri Professor G. Padmanabhan, sir. Please put your hands together. It's a great honor for us to felicitate one of the most celebrated scientists of the country, who is also renowned across the globe. Thank you. Thank you, sir. May I now request Professor Deshpal Verma, sir, guest of honor for today's conference, to please accept our felicitation. I request Krishna Devaraylu, sir, to please felicitate Professor Deshpal Verma, sir. Thank you, sir. We once again thank all our dignitaries. And I think the energy flow in the hall can only be witnessed by a loud round of applause for our dignitaries. Thank you. As we come to the end of the formal inaugural session, may I request Professor D. Vijay Ramu Garu, Dean Foreign Collaborations Vignan Groups, to propose a formal vote of thanks.
Good morning, everybody. I feel very happy that the inaugural session has set the right scientific temper for the 3 days international conference. I am glad to inform that this is truly international conference represented by participants across the globe. On behalf of the Department of Biotechnology, I feel privileged to propose this vote of thanks for this scientific colloquium. Initially, I would like to thank His Excellency Dr. Sir Richard J. Roberts, Nobel Laureate, New England Biolabs, for taking part in this symposium. As soon as I started learning ABC of molecular biology, I became acquainted with the products that NEB has made, like restriction endonucleases, T4 DNA ligase, Tim-Falkline phosphatase. So although I used products of other competitors, but still I felt their products were the best. And then, thanks to Professor Deshpal Verma, the Ohio State University, USA, for which they proudly call, and Dr. Nessi Srinivas, International Research Institute, Philippines, for readily agreeing to give talks spread over 3 days. I would also like to thank the chief guest of the inaugural function, Padma Bhushan Dr. Padmanabhan Garu, former director IISc and chair BIRAC. It is also my pleasant duty to acknowledge with thanks all the invited eminent speakers such as Professor K. Tangaraj, Director CDFD Hyderabad, Dr. Narahar Sastri, Northeast Institute of Science and Technology, Assam, Professor Chandrasekharan, Johns Hopkins University, USA, Professor D.N. Rao, IISc Bengaluru, Dr. Bhanu Prakash Reddy, National Institute of Nutrition, Hyderabad, Professor J.S.S. Prakash, University of Hyderabad, Professor Bipin Nair, Amrita University, Kerala, Professor Nirmal Kumar Lohia, University of Rajasthan, Jaipur, Dr. Geetanjali Yadav, Dr. Pragya Yadav, Dr. Jeevan Jyoti Panda, Dr. P. Ratnagiri, USA, Professor J. Raman, Flame University, Pune, Professor Pushpendra Singh, Madhya Pradesh, Dr. Krishna Mohan Birla Institute, Jaipur, Dr. Chandra Basu, BHU, Varanasi, Dr. Prashant Survajala, Amrita University, Kerala, and Dr. Sugunakar, LPU, Punjab, and Dr. Arifullah Khan from University, Malaysia, Kelantan, and his colleagues. And we would love to thank many delegates who have taken pains to come to VFSTR to present their research data to enlighten all of us. We have received immense support, help, and cooperation from Dr. Lau Rattai Garu, Chairman, Vignan Group of Institutions, and Mr. Lau Sri Krishna Devarayal Garu, Vice Chairman, Vignan Group of Institutions, who is also Honorable Member of Parliament, Lok Sabha. Without their encouragement, it would not have been possible to organize international conference of this magnitude. We cannot forget the financial support rendered by VFSTR for organizing this 3 days conference. We will be failing in our duties if we do not acknowledge the kind cooperation, timely advice, day-to-day guidance, and mentorship from the Vice Chancellor and the Registrar of VFSTR. Honorable Vice Chancellor has asked us to conduct pre-conference tutorials, which were well organized, and it was good scientific exchange of information. Yesterday, it was kick-started. This conference has kick-started with the pre-conference tutorials about machine learning and also next-generation sequencing and its applications. We gratefully acknowledge the kind support from the Department of Biotechnology, Ministry of Science and Technology, Government of India, Andhra Pradesh State Council for Science and Technology, India, Andhra Pradesh, India, Bharat Biotech International, Hyderabad, India, Unipath Specialty Laboratory Limited, Ahmedabad, India, MGI Tech Company Limited, Singapore, Avant Sante India Private Limited, India, Study Orbits, Hyderabad, India, Genomics Call Private Limited, Pulivendula, Andhra Pradesh, India, Vaster Life Science, Bangalore, India, Hetero Biopharma Private Limited, Jedcherla, Telangana, India, Punjab National Bank, Guntur, Hetero Drugs, Hyderabad, Infinity Healthcare, Nellore, SSR Chemicals, Tirupati, Bros Chemicals, Tirupati, and SD Chemicals, Hyderabad, and many others. We immensely thank the BioClues organization for giving us an opportunity to host this international conference at VFSTR. I like to acknowledge the advisor, Department of Biotechnology, Professor Kavikishore, who is the man behind organizing this conference, and Professor Krupanidhi, Dean School of Biotechnology and Pharmaceutical Sciences, and Professor Venkatesh Sorlu, HOD Biotechnology, Professor G. Srinivasrao, Dean R&D, and Professor Pichaya, Head Department of ECE, and Professor Srinivas Babu, Vignan College of Pharmacy, Principal, and Professor Jitendra, HOD Pharmacy. And we would like to thank every personnel for the direct or indirect involvement in making this conference a kickstart in a good manner, very good manner. And finally, I would like to thank all the faculty and students of Department of Biotechnology for bringing this to this platform. Thank you all.
Thank you, sir. Let us now take a short high tea break, and I request all the delegates to please come back to the venue in 15 minutes for the technical session 1.
Good morning, everyone. I welcome you back to INBIX'22, the first technical session of our international conference on frontiers in nutrition, medical genomics, and drug discovery. In this session, we have 4 very knowledgeable lectures by eminent scientists. To chair this valuable session, may I now invite Professor Bipin G. Nair, Dean, School of Life Science, Amrita University, Kerala. Sir, kindly grace the chair. I would like to also invite the co-chair of the session, Dr. Geetanjali Yadav, Senior Staff Scientist, NIPGR, New Delhi. Ma'am, please grace the chair. I now formally hand over the first technical session to the chairs. Ma'am, please take over. Sir, please take over the session.
Perfect. Yeah, works. Yeah, exactly. So I was wondering if Professor Padmanabhan will join us, and should we begin then or should we begin already? We can begin already? He is the first talk, so okay. With Professor Rao's talk then? Is Professor Rao here? Oh, perfect. Perfect. So good afternoon. It is still morning. Good morning, everyone. Welcome to the session, the first technical session. All righty. All righty. Welcome to the first technical session that we have for the INBIX International Meeting 2022. And the first speaker would be, for this session now, would be Professor D.N. Rao from the Indian Institute of Science, Bangalore. He will talk to us on the DNA-protein interactions using Helicobacter pylori. He has threatened me not to introduce him too much, and so he's welcome to please take the chair and start speaking.
This is good enough. Yes, sir. Hello. Yeah. Yes, sir. Okay. You have a project. And the pointer? Pointer, yeah. Okay.
Professor Rao, you will have about 20 to 25 minutes to speak. We will give you a little warning shot at 20.
Yeah.
And then you have 5 minutes,
Yeah.
and then Q&A 5 minutes.
I can cut it down
Perfect, that'll
whenever
be great.
you tell me.
That'll be great, okay.
This volume is a bit too much. I think it's okay. All right. Can I have the first slide? That's the second one. All right. Good morning, and first of all, let me thank the organizers for having invited me to give a talk here at this very interesting conference on nutrition, medical genomics, and drug discovery. Yes. All right.
I'm sorry to interrupt you, sir. All the participants are posters 1 to 50 are requested to go to Convocation Hall for poster presentation and parallel sessions. Thank you.
What I'm going to speak to you this morning is on a very interesting organism called Helicobacter pylori, which I think is a very harmless organism, but it becomes virulent or pathogenic, then you have is a causative agent for ulcers, gastritis,
And it's the first known bacterium which causes cancer. The gastric cancer is because of Helicobacter pylori. The Helicobacter pylori is a very interesting organism. And I'll tell you some history about the organism because that is important. And as you go down, yes, this is where Helicobacter becomes very important. Well, all of us know from history that Alexander the Great conquered from Greece all the way to this portion of the world. The Helicobacter is all over the world. In all the continents, at distant places, you have Helicobacter, the empire of Helicobacter pylori. And more interesting about the Helicobacter pylori is the migration of this particular bug. And you can trace it with actually the migration of human beings, the humans from Africa all the way to South America to Australia and things. Both the evolution of man, or the migration of man, and the migration of Helicobacter pylori absolutely coincides and superimposes, and this is what people have studied. Now, one of the interesting things, I put this slide is only for the students, that about 4 years back, there was a report that came out in Nature where they found that in the the Tellurian Alps of Italy, they actually found a body. And inside the body, when they opened up, it had, in the stomach, they could culture the Helicobacter pylori that is present in this man, who was 5,000 years ago, he actually was killed by an avalanche. He was actually murdered and then buried in that avalanche, and then they picked him out. And what became interesting was, if you look at the Helicobacter pylori genome, when the sequence was done, it looks like his ancestors inherited bacteria from Asia rather than from Africa, because all of us know it's out of Africa that things have started. But here is the first linkage, first discovery, first link to an Asian ancestor of Helicobacter pylori. So the bug survives at very acidic conditions in the stomach where the pH is 1 or 2, and has a very good number of strategies to survive this very harsh environment that it's present in. It's a gram-negative epsilon proteobacteria and causes, if it's not avirulent, if it becomes virulent, it's associated with a number of gastric pathologies as listed here. Now, Helicobacter pylori got importance for a number of reasons. I'm trying to get this done. I'll skip some of these slides. It has infected with the humans, and when the human evolution started 58,000 years ago, the first traces of Helicobacter also was traced to this. Jaworski at Krakow University at the turn of the century, last century, 1900, saw something like this spiral-shaped organisms, or helical-shaped organisms. And it's about 83 years later, or 84 years later, two Australian scientists, Barry Marshall and Bob Warren, actually showed by doing some very nice experiments, and I'll tell you what the experiment was, that Helicobacter was the causative agent for the ulcers and gastritis. So it took 86 years before the Australian scientists identified Helicobacter as the causative agent. In the stomach, you have a number of organisms, right? We have, all of us have 100 trillion bugs there, but there's a related organism called Campylobacter jejuni, which just looks like Helicobacter pylori. And it's only because of Marshall's and Bob Warren's experiments that Helicobacter pylori was identified. And one of the two scientists there, Barry Marshall, actually drank a pure culture of Helicobacter pylori and got himself gastritis, and then proved the postulate that this is the organism, the pure culture that he drank was caused for the gastritis. So that is how this was finally, the whole reason for gastritis was because of Helicobacter pylori. And a lot of nice work from both these scientists, Barry Marshall and Rob Warren, then led to the discovery of this as a causative agent. And then they got the Nobel Prize in 2005 in medicine or physiology for the discovery of this particular bug. And this is the two of them celebrating the Nobel Prize. This is the bug that is present, as I said, in all of us, is harmless. 60% of the world population harbor the Helicobacter pylori. So most of the population of this world has Helicobacter pylori. It's a harmless organism, but if once it becomes virulent or pathogenic, then you have gastritis, you have ulcers, and then it's also a carcinogen which causes cancer. So I thought I'll tell you a bit more about this bug and then to come back, if time permits, what kind of research that my laboratory is actually doing. Now, one of the most important things that, which I want to tell you about this particular bug, that it dominates in the environment of a very harsh nature, acidic nature, where the pH is 1 and 2. And the bug is not an acidophile. It's not that it grows on acid. It actually uses this enzyme called urease, which degrades urea to ammonia and carbon dioxide. And the ammonia in turn neutralizes the HCl that is present in the stomach, and that is how the bug survives at pH 7. So it's not an acidophile, it just has a very nice strategy of overcoming this acidity by the enzyme urease that is present in the Helicobacter pylori. Nothing moves. Hello, can I move on to the next slide? All right. Can you help me if I say next slide? Maybe that is better. Right. Now, one of the most important things about Helicobacter pylori, it's a very diversified genome it has. Even in your stomach, you have regions 1 millimeter away or few millimeters away that the population of Helicobacter pylori is a different genome completely. So the diversification, the diversified genome is a hallmark of this particular organism, and because of that, and because of this high frequency of genetic recombination, you have this polymorphism that you see in this particular bug. So there's a high mutation frequency, there's a presence of error-prone DNA polymerase, makes all the mistakes, and therefore the bug survives. There's something called phase variation, there's epigenetic diversity, there's an intragenomic recombination, the highest of in a known bacterium, and the bug is naturally transformable. You just have to show DNA around it, and the DNA gets in. Now, this is where the interest in my lab came, that Helicobacter pylori, the genome was sequenced in 1997, 98. 2 strains were actually sequenced, and they found that the bug harbors a lot of restriction modification enzymes. You remember what Rich Roberts mentioned this morning. The restriction modification systems are basically defense mechanisms. If a phage comes in, a foreign DNA comes in, the host DNA, the host will chop off the foreign DNA by using restriction enzyme. And that's the purpose of the RM systems. So the Helicobacter pylori genome, although it's a 1.5 MB genome, harbors 40 to 44 restriction modification open-reading frames, enzymes which resemble restriction enzymes. And that is where my lab got interested in 98, 99 as to why was this bug having so many restriction modification systems, especially when it harbors in your acidic condition of your stomach where there are no phages at all. So what are the RM systems doing in this genome? And therefore, there must be some other reason why the RM systems are present. It may not be just for genome, a phage protection, but the phage destruction, but it has some other roles. And therefore, we started working on RM systems, but I will talk a little about that because there are more interesting features of the Helicobacter pylori genome which are interesting to talk about. Another thing that I mentioned was Helicobacter pylori is naturally competent, and therefore the transformation frequencies are very high. And therefore, if you now see the transformation pathway, and we have studied some of the details of this pathway, you find that, the DNA, the competence is very high. The DNA binding and uptake mechanism have been very well worked out here. We know all the proteins that are involved. This is not my work, it's work from other laboratories. The DNA is processed, double-stranded is converted to single-stranded, there's homologous recombination, and then you have the recombined DNA. And this is where the paradigm is that, the bug carries so many restriction modification systems, and the whole purpose of RM systems is to cut foreign DNA. But yet, foreign DNA still comes through and then goes through this process and gets recombined. So there must be other reasons why the restriction enzymes are present there. And that is why the laboratory is interested to find out what is the role or what are the other roles of the RM systems other than the defense mechanism that people now normally associate with this particular phenomena. So we have studied the natural transformation pathway in Helicobacter pylori and the RM systems. I'm going to tell you very little about both of them because we have other things to discuss about. Next slide. This doesn't work. All right. This is slightly an old slide where only 4 Helicobacter strains were sequenced. Now we have about 200 strains that are sequenced. We ourselves in Bangalore, in my laboratory, have sequenced about 10 of them, mostly Indian isolates. And we find that, many of these genes, in all of them actually, in fact, the RM systems are in large numbers. At least 1 to 2% of the genome genes are the RM systems. Of course, all the other virulent genes are present here. This is just a comparison of 4 strains which have been completely sequenced, and to highlight the point that each strain has specific RM systems, and that is the characteristic feature of this H. pylori. It mutates heavily and it keeps on changing, and therefore the question that one needs to ask, and I think the students of the future, people who work on H. pylori should ask, why isn't that genome disappearing? If it's all the time mutating, somewhere along the bug should disappear, but it has not happened at all. So we like to study what are the mechanisms that keep H. pylori, keep on growing, keep on mutating, and still survive in the acidic pH condition that you have. Next slide. All right, I'll skip this slide. Next. All right. So this is what we did, just to show you the Indian isolates of that. And I think one of the most interesting features that came out from our genome study of H. pylori strains, that while most strains that were sequenced by the Western laboratories have about 44 open reading frames that look like RM systems, we in the Indian strains have much lesser RM systems. There are about 30 to 32 RM systems. So that's an interesting thing to pursue, which we are not doing at the moment, to find out why do the Indian strains have much lesser number of RM systems. Still, we don't know the function of these RM systems because the difference hypothesis goes nil here because there's hardly a phage or two that invades the stomach. So we're still interested in that. So this is how the bug looks at. It has 2 to 6 flagella. The flagella can exist at the 2 ends during fission, and the flagella plays the more important. So all your virulence genes, for example, are all the flagella genes, for example, that are very characteristically present. This is the morphological thing. Helicobacter pylori is now becoming, also important to study because it forms biofilms, and therefore the resistance to a lot of drugs that normally people take for H. pylori are not working at all. So there's a need to study the biofilm mechanism and how to avoid biofilm by taking small molecules and things like that. But we're not doing that kind of work. But I think this is important now, like all many other drugs where drug resistance is now becoming a major feature of research in many laboratories. Next slide. All right. Next slide. I'll skip this. That is the enzyme that I was talking about, and these are the virulence factors: the urease gene, the flagellar genes, the exotoxins, the secretory enzymes, the type 4 restrictive system, and the effector molecules, that the KAG and things like that, which actually are secreted and go into and infect the gastric cells, and then there's inflammation, and then therefore you have cancer and things like that, which go on as a part of this infection of this. Next slide. So the outcome of infection in general is because of three factors. That is the strain virulence, the environmental factors, and the host genotype. It's a complex interaction between these three features that will tell you whether the H. pylori is going to successfully infect and then survive in the particular disease condition. Next slide. Next slide. Next. All right. So these are the diseases associated with H. pylori. The ulcers, both the duodenal, the stomach. And if you go through, this is where it comes that, the next slide, you can go, sorry. This is the stages of dyspepsia to chronic gastritis, all the way to carcinoma. And by this point of time, there's no return. This becomes very dangerous for the organism to survive, a human being to survive. Let me try this. I want to show this because we actually go to the hospitals to take out gastric, the ulcer samples. Actually, from here we isolate the bug and then purify it, and we have a pure culture of Helicobacter pylori, sequence this. So there's a lot of involvement with the clinicians that we get into, and therefore it's been very, very difficult to sometimes to isolate these or get these samples just to do these kind of experiments. All right, let's skip that. So my laboratory has been working on H. pylori since the time it was sequenced, about actually 5 years later on, and we have studied mostly interaction of proteins with DNA. So we not only study on the restriction modification systems, which are shown here, we also work on the natural transformation pathway and on the repair pathway, which I'll show you in the next slide, which is here. So this is where we're talking about recombination, where in H. pylori we have studied DNA topology in terms of studying the topoisomerase, which is important. And more recently, my last student, my 25th student, worked on protein acetylation in Helicobacter pylori. My laboratory does mostly biochemistry of these things, biochemistry kind of work. So we have purified enzymes, studied these enzymes, and in some cases we have actually done some transformation studies to find out about the mutant strains and things like that. I'll briefly, very quickly go through some of this work here. As I said, the H. pylori has a number of RM systems, 42 to 44 of them. I couldn't put all of them in one, therefore they're all shown as you have these bars that show that you have. Oh, terrible, 2 minutes? I just started my talk. Anyway. All right. So we work on the RM systems, and I thought, I'll just go through the next few slides quickly. It is one of the organisms which has the maximum number of RM systems compared to the other ones. And this is the Indian strains. We worked on the methylation, the DNA methylation. Very interestingly, we found a methylase which actually methylates two methylases methylating two strands of the same DNA molecule, the upper strand and the middle. So we have studied the enzymology of this. Very interesting thing that we have done with these enzymes. Next slide. Oh, this is terrible. Yeah. So we actually, in collaboration with Ashish Mukhopadhyay at the Calcutta NICED, we went about looking for these genes of this methylase in symptomatic and asymptomatic patients. The tribal population, the Andaman Nicobar Islands, who have H. pylori but have never come down with any gastric infection or ulcers and things like that. So we looked at strains of this, looked for this particular gene, and we found some very interesting correlations. Lack of time, I'm not going to mention the details of it. Here's the methylase. I'll stop with this. That it's actually split into two open reading frames, one called the 1369 and 1370. And there are 10 Gs in between. This is inactive, this is inactive. But the minute you introduce the 11th one, you get an active methyltransferase. And that is what the H. pylori has devised all these kind of strategies either to make them non-functional or functional at various points of times in the life cycle. So we have done, Ritesh in the lab actually did some of this work, and I have to skip many slides now just to tell you that this is a very interesting organism. I want to talk to you about this, but I think, we have now used the single molecule real-time sequencing, SMRT sequence, to find not the sequence, but only all the methylated bases that are present in the organism. So SMRT sequencing has been very, very helpful, and this was done in collaboration with J Roberts at the New England Biolabs, and this is the system we have actually studied. Sorry, I have to skip this, and I'll come back. This doesn't work. Oh, so to end the talk, that RM systems in H. pylori are dynamic. They're born, they're maintained for some time, and then they die. So all the time in the system, you dig into the stomach, get H. pylori, you can get the new restriction system. And that is how the bug survives under harsh conditions, to make mu and neo molecules which help it to survive in this condition. So I conclude saying that this is a dynamic, a diagram showing dynamic acquisition and loss of RM systems. And one of the things very peculiar to this organism is this particular change of specificity during the lifetime of the bacterium in the gut. So I'll stop here. Many more other things to talk about, but thank you very much. And we have time. Otherwise, I'm here for 2 days and you can have questions.
Thank you, Dr. Rao, for that interesting talk. Time for a couple of quick questions. Maybe you can.
Yes, sure.
Another organism that has high level of competency is Vibrio cholerae, which can take up the outside DNA very efficiently. So how different it is from that in terms of its mechanism of taking up DNA from its environment? How different the mechanism of taking up the DNA from the environment is between the Vibrio cholerae and Helicobacter pylori?
Good question. Vibrio cholerae, Agrobacterium tumefaciens, and Helicobacter pylori have that chart that I showed you, uptake, processing, is very similar. Many of the proteins are similar to all these three systems. But the only uniqueness about the H. pylori is that it takes up both double-stranded and single-stranded DNA. And whereas in the others, it's just the double-stranded DNA and things like that. But ultimately, even the double-stranded DNA is processed as single-stranded, and then that is integrated into the genome. So the mechanisms are about the similar. The proteins are similar, not in the sequence, but the functions of these proteins are similar. There's a protein called DPRA, DNA processing protein A, which is common in all these proteins. So that is the most important one to take the DNA, the uptake of DNA.
Vibrio cholerae doesn't have the same high level of genetic diversity.
Yes, that is true. Vibrio cholerae doesn't have, even, although the transformation, as I said, the proteins in the mechanism are about the same, but the DPRA, for example, is very, very efficient in this case, in the case of Helicobacter pylori. You have a similar protein in Vibrio cholerae and in Agrobacterium tumefaciens, but I think the level of transformation, efficiency of uptake is much less.
Thank you very much.
Thank you, Dr. Rao. One more question.
Yeah.
Here.
Yes.
Sir, it was a very nice presentation. So my question is, there are lots of companies are there in the market who are working on the gut microbiome, and they are giving some kind of reports along with some medicines are also there. But when I talk about the your presentation after listening to you, so it is definitely one of the most important factor this H. pylori can give to any individual. So, and population wise also, you have already mentioned like it is different in different population, it behaves separately. So what do you think, sir, the research of this H. pylori has reached to a stage that because of which any drug can be decided, or it can be, or it is already there. I don't know about this. So
No,
I'm asking this.
I don't think there's any specific drug at all. It's a very general drug that, and it's usually when you have H. pylori infection, the clinicians or the doctors prescribe proton pump inhibitors and things like that. It's a very general thing. There's no specific thing like that.
Okay.
But if you look at the population and you look at the genome sequences, there are variations. Variations probably not in terms of drug resistance or things like that, but it could develop into a drug resistance. But other kind of things have developed, you know, for example, this system, this bug doesn't have a mismatch repair system at all. So no post-replication repair system occurs at all here, and therefore the mutation frequency is high. Addition, for example, in the sense, you know, the pylori infection, the CAG expression, the CAG, the VAC expression, they keep varying because of the mutations and have. Infectivity or pathogenicity varies because of these mutations. So some are tolerable, some are not tolerable at all. But in terms of drug resistance, there's hardly any difference. And the people have not noticed, or people haven't identified anything like that so that you can target with one particular drug and things like that. Most of them are proton inhibitors that that are prescribed by clinicians when you have infection.
Thank you so much, sir.
Yeah, in the interest of time, Dr. Rao is here till tomorrow, so please consult with.
May I now request our chairs to kindly felicitate our speaker, Professor D.N. Rao.
You know, when you raise it, and that's a better background, rather than making it stand in front of the screen. That takes our time. Let's do it together at the end.
No problem.
Before we move on to the next lecture, I have an important announcement for the participants of poster session. The poster session for posters 0 to 50 is going to take place as per the schedule in the afternoon, but the candidates are requested to fix their posters during the lunch time for to felicitate the inauguration. Thank you. I hand over the session to the chairs now.
Thank you once again. We now have as speaker Padma Bhushan Professor G. Padmanabhan, whom you had heard in the morning on a general speech, but now he will be talking to us technically. He is currently the Senior Science and Innovation Advisor at BioRac and will speak on the contours of biotechnology in India. We have a scene here. We are not giving an introduction to each speaker, and I encourage all of you to open your abstract booklets and check out who we are, where we are from, and what we are doing. But about Professor GP, as we like to call him, I need you all to know that he is responsible single-handedly for coordinating various committees in the country, particularly within BioRac, BIG, SBRI, and so on. Just to see an example, you see one scientist in him out of over 1,000 scientific innovators who's developing orchids. Professor Padmanabhan, over to you now for your technical talk. And we will give you 15 minutes because we know people will have questions. So 10 minutes for Q&A followed by 15 minutes of your talk. If that's, we'll give you a buzzer when 2 minutes are remaining. Please, you may begin now.
Can I have the first slide? Yeah. Screen, full screen. I have to thank a lot of people, but because of time, there's only one name I will take it, who epitomizes the whole Kavi Kishore. He is the one who quietly is at the back, but he has brought everybody together, and it's indeed a great privilege and pleasure for me to be here. And of course, the other one, the front end, my good friend Giri Pallavarupu, whose products we released in the morning. And all other people, Jalaja, for example, the whole student community is after us. When I said after us, taking care of us. So it's a great pleasure and honor for us to be here. Now, I work on with the malaria parasite, but I also gave the option that I can talk about biotechnology development in the country because I've been associated for the last maybe 4 decades. Even before the Department of Biotechnology came into being, I've been associated. They chose wisely the second topic for the reason, research, only few may be interested, but there are a lot of young students with all the startup culture coming. Maybe, if they listen to you on the contours of biotechnology, maybe they will have some ideas on that. So it's fine. I will talk about that. Next slide. I think better you can move the slide then. Or shall I do that? Just 5 minutes on my research. I have spent in my entire career 60 years of science in this country. So, with perhaps 2 molecules, and one molecule is the heme. The heme is the prosthetic group, as you know, of hemoglobin, all cytochromes and so on. But we asked a different question. What does heme do to the apoprotein? Transcription of the apoprotein, translation of the apoprotein, transport of the apoproteins. So we spent, we started in Neurospora as a fungus, then went into animal systems, and then went to malaria parasite. Why malaria parasite? Heme is a very central molecule for malaria parasite. In the blood stage, it is living in a sea of hemoglobin. And it breaks down hemoglobin and all the heme. What does it do? It's so toxic. The parasite forms a polymer called hemozoin. Which is the inert molecule, but so far as parasite is concerned, but it is very detrimental to us actually. But our exciting finding was the parasite makes heme. It has all the genes, all the pathway. We spent a lifetime cloning all the genes, etc., etc. I will not get into it. So this question has remained. It's living in a sea of hemoglobin. Why does it make heme? And this has been, we have the answers. And one of the things is it is required for the mosquito stage. You remove the heme, you make knockouts of the genes and so on. The parasite grows merrily in the blood stage, but it will not grow in the mosquito stage. So we published it in pathogens, etc., etc., all that we do. But what is more important is, I was still asking this question, why does it make it in the blood stage when it is required for the mosquito stage? Then we found it is required for virulence of the parasite. If the parasite does not make heme, it doesn't cause cerebral malaria. That is something very exciting. We have published it in Nature Communication very recently. When I say we, it's all done by Dr. Arun Nagaraj at Institute of Life Sciences, Bhubaneswar. They were all part of my group. Now I am part of their group. They are the leaders, but I am a sounding board. It keeps me kind of engaged. We have Skype meetings every week, twice, or do that. So very exciting. This is, we thought, if the parasite, how is it able to distinguish between hemoglobin heme and its own heme? We still don't have the answer for that. We're searching for that. But this heme is so particular for cerebral malaria. So we thought if you have an inhibitor, because you have done knockout, etc., etc., that's all right. But can you get an application out of it? So, next slide, please. We found a molecule. This is griseofulvin. It's an antifungal molecule, actually. But interestingly, it inhibits heme synthesis in the parasite, and it inhibits cerebral malaria. And this is a repurposed drug. It is actually an antifungal, but we are now showing it is useful to cerebral malaria. And hopefully, we are planning to do clinical trials, etc. Because a lot of data, a lot of people are prescribed for this antifungal infection, this molecule, griseofulvin, and they take it orally for a long time. So a lot of safety data is already available. So we think we will be able to take it for the clinical trial. That's one project. The other one, next one, please. This is what Giri was talking about. The other project I was interested in is natural products. For some reason, we claim, we have lot of natural products, but we really have not taken them forward, to their application. But at least, then that's how, it's a big history, but that's how I came down to curcumin, turmeric, and then curcumin. Next slide, please. Let me, I have few slides on curcumin little later, I will come back to it. But for the students, the term biotechnology was previous, right? By this guy, Carl Ericke, the Hungarian engineer who coined this word biotechnology. Next slide, please. I must confess, I will be very superficial because the field is so vast. I can probably, sometimes only give you the title, but at least you can go back and see what does it mean. For example, synthetic biology. I don't have time to explain to you synthetic biology. But you should go back and see what is synthetic biology is all about. That is what I'm putting right in the beginning. Okay, the biotechnology is because of all this. I'm concentrating biomedical sciences, biochemistry, microbiology, genetics, molecular biology, immunology, medical sciences, neurosciences, engineering sciences. All these are behind biotechnology. It's not one field actually. You may be studying it as a course, but all these fields are behind this technology. Next slide, please. Now, many revolutions have taken place. Genome revolution, I will not talk about it. But just one point I want you to remember, in my career, I have cloned 20 genes, 25 genes. Today, cloning a gene is a child's play. In our time, there was nothing. We will start with the protein, make antibody, immunoprecipitate the polysomes, isolate messenger RNA, convert it into cDNA, clone it. No kits were available. A big deal. But once genome sequence came, who cares? They just go to the computation and then identify the genes. You all see a top-down approach. This is what this is trying to tell you. We all had bottom approach from the protein to the RNA to the gene. But now you come from the DNA down you come. You know the sequence of DNA. So, but I tell you, biochemistry, I'm professional biochemist, I have to defend my field. It's still very important. You can have the gene, you can have everything, but already, what is the function? You have to do biochemistry. But the point is, it has become a top-down approach, and all these both are used for drug targets, vaccines, pharmaceuticals, diagnostics. And these are the areas I will be very briefly telling. Next slide, please. Now, before that, just, my talk will be, because of topical relevance, COVID context. Because I chaired the committee for vaccine development for COVID. So I am fairly up in this, what is happening, at least technically. This is a virus, RNA virus, about 30 kilobases, slightly less, 29,000 something bases. And about 25 non-structural proteins and 4 structural proteins. And everybody talks about this spike protein. But what I'm telling, there are other proteins probably are more important or maybe become important in terms of diagnosis, perhaps at some point of time, or in terms of therapy. This is what I feel. It makes a polyprotein of 16 proteins. And of course, they're all cleaved, and then where you get all these proteins, and then you have the spike, you have the envelope, you have the membrane, you have the nuclear capsid. Structural proteins are there. Next slide, please. And we all know how the virus enters. It binds to the what is called the ACE2 receptor, and then it gets inside the cell through the receptor in the host. Lung cells are supposed to be very susceptible. And then the virus gets degraded, and of course it will again code back and then reassemble, and then the virus particle comes out, multiplies. That is the cycle, and this is where you can see the spike protein, and the yellow thing is the ACE2 receptor. Just lock and fit, very, very fine fit that you see. Next slide, please. So I just gave you 2 slides to say what basic research is all about. Before the disease was understood, SARS-2 or COVID, the sequence is out, what all it does, I mean, all this been information, technical information is available, except we don't know how to control this virus. This is, I keep saying this. You know, we think we have so much knowledge, we can do this, we can do that, but this virus has its own mind. I have seen so many mathematical models. Oh, it will go up like this, it will come down like this. None of the models prove to be right. This virus is a very smart virus. I somehow feel, you know, we still have challenges. It only gives you a sense of humility. You know, it's not as if we know nature. Nature has some secrets up the sleeve all the time. I somehow feel. Next slide, please. Now I don't know whether I should claim credit or not. This letter I wrote in Nature Medicine in 2003. I just see the concluding sentence. In short, India has the scientific capability and modern infrastructure to eventually assume leadership in global vaccine manufacture. I said that. And many people quote these foreigners, you know, when they correspond with me. You said this, or your country is proving it to be right. I'm just telling you, I mean, I'm not a big, but India really has leadership in vaccines. This is few slides will tell you. Next one, please. Viral vaccines, polysaccharide vaccines, recombinant vaccines, combination vaccines. As I said, I'm superficial. I don't have time to get into deeper. For example, we made a recombinant hepatitis B vaccine in our lab, Professor Rangarajan, in our lab, we made it. But only after Paraprasad Reddy in Shanta Biotech made it. He was the first one, and he put India in the global map. It was his hepatitis B vaccine that put India in the global map, actually, recombinant hepatitis B. We did 4 years after him, but we also gave it to 3 companies. We gave it to Bharat Biotech, we gave it to Indian Immunologicals, we gave it to another company. All of them are making it, actually, recombinant hepatitis B vaccine. Of course, combination vaccines, now you talk about pentavalent vaccines, hexavalent vaccines. You can't have individual vaccines and poking the child 20 times. So you mix 5 vaccines, you mix 6 vaccines, and then give them. Next one, please. Well, in SARS-2, what we have done? Covishield, before that Covaxin. Covaxin is Bharat Biotech. In fact, I remember in the first meeting I told you I chaired it, I raised this question, you know, classically we used to give the organism that produces the disease. We used to inactivate it, chemically inactivate it or genetically inactivate it. What happened to the technology? Oh, somebody said it's all old technology, who will use it? But then, you know, here comes Bharat Biotech, it's only inactivated virus. Of course, they use, the cell culture is not very conducive, you know, it grows very slowly, I know that. But they collaborated with the ICMR and they made this Covaxin. This is just the inactivated virus. The problem is with the yield of this virus growing in cell culture. So, in fact, even today, Bharat Biotech will supply only 15% of the total vaccine requirement. Everything else is supplied by the Covishield. Because the yield of the virus is low. That is the challenge actually, how to do that. We have to still see. Then comes the Covishield. This is the adenoviral vaccine vector carrying the S gene S gene. We all know that this is AstraZeneca product which is now made in Serum Institute in India. And we have this non-replicating 85 carrying S gene that is I must tell you, all the vaccines are made against S gene, the spike protein, or the RBD, the receptor binding domain. This is also part of a spike protein. So that is why I said there are other structural proteins. Maybe people should, I mean, there are a few who are looking at it, but nothing has come out still, in my opinion. Then interestingly, we have DNA vaccine. In In fact, we made DNA vaccine against rabies 3 decades ago, but I must tell you, not a single DNA vaccine is for human application, even globally. The only DNA vaccine application I know is for an West Nile disease in horses, not really in human actually. But for the first time, India, I think Cadila, they have got DNA vaccine for COVID, SARS-2 virus, which has been approved for human use. But they have a, problem. You won't see much of DNA vaccine in the market because the yield is a problem, making it the pure DNA is a problem. The other one is the mRNA vaccine. Again, I should say, look, I told you earlier, maybe I worked with 20 mRNAs, whatever. Never thought mRNA will make a vaccine. That is where innovative thinking is about. Somebody thought, yes, mRNA can be a vaccine. And we all used to think mRNA is unstable, you cannot use it for any application. But then this lipid nanoparticle, coating it with the lipid particle, etc., make it stable. So we all know the 2 companies which are making the mRNA vaccine. And India, I must tell you, Genova at Pune is making mRNA vaccine. Has made mRNA vaccine, approved by DCGI. The other day he called me, he said, sir, government won't buy it. It was very unfortunate, I can tell you publicly, the government encouraged them to do mRNA vaccine, and when they have made it, they are no buyer, they are a little worried actually. And their mRNA vaccine is very interesting because, the Moderna and all that, they need minus 20 degrees, minus 80 degrees for storage. Genova's mRNA vaccine can be stored between 2 to 8 degrees. It's a big breakthrough they have. And it is a self-amplifying mRNA. I don't have time to go into the great. Please look at what is self-amplifying. That means it has DNA polymerase and RNA polymerase. The RNA that is formed will be amplified. That is where. So it is a very novel kind of thing with this Genova is made, but I hope they will be able to. They are now looking at export market and so on. And so on. Next one, please. Now this is briefly for the students to know what is the DNA vaccine, what is the difference between a DNA vaccine and an mRNA vaccine. A DNA vaccine, you have, we got the gene, spike protein gene, which is introduced into the cell. I told you how the virus enters like that. But then the DNA has to get into the nucleus. That is the one limitation. DNA has to become into the nucleus, then converted into RNA. RNA has to come out, and then in the cytoplasm you have to get the protein. Whereas mRNA vaccine, in the mRNA, directly gets into the cytoplasm and then translated. So there is no need for entering into the nucleus and all that. That is the biggest advantage of mRNA vaccine. So both these vaccines have become very popular in this COVID era. Next one, please. Next slide. So what is the Indian effort? I told you already, we are making mRNA vaccine as of today. DNA vaccine, recombinant protein, I think right in Hyderabad, the biological events is making spike protein, recombinant spike protein, or the receptor binding domain. Of course, there was also other, whether recombinant BCG, why Indians get less of COVID. That was one. Maybe we are all vaccinated with BCG when we were young. So we carry some immunity in us, all kinds of things were said. But so projects were supported, even for making BCG, whether, it will help against general non-specific protection against COVID. Of course, there are other platforms I should tell you. I told you about the adenoviral platform. People are also looking at rabies virus platform, influenza virus platform. Now, next slide. You can go next slide. Yeah. So there are vaccine challenges in India. I must tell you, India exports vaccines to 150 countries. And every third child in the world gets Indian vaccine. pediatric vaccine, if you look at every third child in the globe gets an Indian vaccine. So this is something which can be proud of. So combination vaccines, cold chain, we still haven't solved the problem. Vaccines have to be stored at 2 to 8 degrees or even lower. Those conditions, large country like ours, when the temperature can go to 45 degrees, now how do you store these vaccines? They are all challenges. Of course, pneumococcal vaccine. Delivery systems is very, very important. Viral like particle, VLP, oral, nasal, dermal patch. These are all areas of research. Should you be poking the child with the needles all the time, or alternate routes of administration, dermal patch and so on? These are very exciting opportunities for you to look at. Then of course cholera, tetravalent, dengue, influenza. Bharat Biotech is making many vaccines, I should tell you. One of the challenges for vaccine, SARS-2 virus, I am sure experts are here, is the mutants which you are getting, variants you are getting, lineages, sublineages, or the vaccines protecting, not protecting. You get all kinds of papers, including Nature. And somehow I have fed up with very low quality papers got published in big journals because they are on COVID. So, but this is the challenge actually, whether the vaccines protect against the variants. Virus will keep on mutating. You will be making a vaccine for every mutant? These are some of the challenges. Whether the vaccine, you can make a universal vaccine, there are attempts although globally. So, vaccine is one part I have covered. I am just quickly going on to biopharmaceuticals. I am just covering the broad spectrum of biotechnology. Biopharmaceuticals, what is the difference between a biopharmaceutical and a pharmaceutical? Pharmaceuticals are generally small organic molecules. Biopharmaceuticals are proteins. Even DNA, RNA these days have become drugs. So these are all the difference, and then examples I've given: insulin, erythropoietin, serum albumin, GSF, streptokinase, interferon. Sorry. The biggest market is monoclonal antibodies for the country to really go for is these monoclonal antibodies. But I must tell you, India is a major global competitor in all this. Next one, please. I have just given a list of biopharmaceuticals which India is making. Pardon? Last slide maybe, or 2 more slides maybe. Oh gosh. You are giving me 15 minutes or what? We've had 25 minutes already. 25 minutes. Yes, it's all right. Please carry on. No, I don't think I can do it. I thought I have 45 minutes or something like that. No. Next slide, please. That puts me a spoke. Okay, I thought this is just useful to the people. Anyway, okay, India is also following this approach of phage library. Can we construct a phage library? We already have a phage library constructed. Next one, please. Now, this is how do you construct a phage library, which is a phage library for antibodies. Phage library for all antibodies. And then you screen this phage library against S protein. And then pick up. First round, second round, these are called as panning. And then you have finally what you do is when you have selected, you isolate the gene, that is the light chain and the heavy chain, and then clone them and express them. That means you are not doing this hybridoma technology anymore. This is all by recombinant DNA technology. Thank you so much. So all by recombinant DNA technology, we make the monoclonal antibody. Next slide, please. No, I only covered two areas, that is the vaccine and biopharmaceutical drug discovery. I must honestly tell you, we were once projected as capital of drug discovery in this country, but we lost the opportunity because we thought we can do generic molecules, that is molecules which are out of patent, you can make it much cheaper in India. So we shifted to that and lost our lead in drug discovery. Next one. For example, when you come to COVID, you don't have a drug for COVID. It's starting from hydroxychloroquine to every other Chinese traditional medicine. Every molecule in the cupboard has been tested. Next slide. Natural product, next one. Next one, natural products. Curcumin, curcumin I got interested because as of 2016, we have 10,900 publications. We have 1185 analogs, 176 curcuma varieties, but not an approved allopathic drug for any ailment. And not at any disease, no disease has been left out. It has been used to test. That was the reason why I got into this. And then I showed it in malaria, at least it works. I will, next slide, because of time she pointed out, I will not get into it. Next one. Next one. Next one. Next one. Next one. Now this gives you the mechanism. Can you just go, mechanism, how does the curcumin act? Basically it's an anti-inflammatory molecule. It just gives you, if you have to have a combination of curcumin and heart ether. You can completely cure cerebral malaria in the animal model, and we have taken it to clinical trial. Next slide. Next slide. Previous one, so I wrote an article, Nobel for artemisinin, curcumin is waiting for ages. I just want to fight out this case because this is something which people have been using all along. And in between there will be one, curcumin causes, turmeric causes this, so that's why. I am just fighting still this game. Next one. This is curcumin, I don't know. There are so many mechanisms have been proposed. Next one. I had an argument going on with the international community. Curcumin may define medicinal chemists. They said curcumin is not absorbed. Curcumin is poorly absorbed. But all this is all, now if you use curcumin along with piperine, you inhibit phase 2 metabolism and curcumin accumulates in blood. You identify enough quantity of curcumin. Next one. I want to take this concept, curcumin from turmeric as a universal adjunct drug. I don't think it should be a main drug, but it should be an adjunct drug. Next one. Next, diagnostics, yes, DNA to RNA to microRNA to non-coding RNA to protein, all have become diagnostic tools today, and you have kits for various diseases. Next one. I'm spending only 10 seconds per slide, okay. Interdisciplinary active collaboration, engineers, biologists, clinicians. This is some exciting field today. In fact, in Biorex sitting there, I find more engineers are applying, more physicists are applying than biologists themselves, actually. Next one. Next one, all this. This is the diagnostic tool, and this is RT-PCR. Everybody would have heard about it. There is a joke. It's bit of time, I should say. The lady forgot to add salt in the sambar, it seems. So everybody in the house tasted the sambar. It doesn't taste. They said, go for RT-PCR. That's what had happened. Next slide. So devices, Biorack has supported many devices, retinal. I have given a huge set of examples. Next one. I must tell you, Biorack has commercialized more than 140 commercial products. Most of them are devices and diagnostics. That is where, whether we can get drugs, whether we can other things we have. Next, they are all examples. Additive manufacturing, 3D printing, a lot of projects come. I'm saying this because students are around. 3D printing is becoming more and more popular in biology. Bio-ink is, what is bio-ink? Next one, I think I have given you there probably. Bioprinting, bio-ink is a combination of living cells and a compatible base like collagen, elastin, gelatin, hyaluronic, silk, alginate. So this is, very, basically what they want to do is a computer-aided design. You want to have an organ, a liver for example. Now can you make a liver in the test tube? That is the challenge. And a company in Bangalore has made it, Pandarum. And that, of course, still not, I should say, far away from a real liver in the sense, of course, press goes and then says all kinds of things, or you can organ transplant, no. But at least drug screening it can do. You can have liver pieces and screen drugs. Next one. Artificial intelligence has come in a very big way in all these projects. I think students should, this is the opportunity. Next one. Big data analytics, India with its strong in IT sector, I think it's a very, very important area. Next one. It's not only DNA analysis, genome analysis, it's also public health of great interest. We do not know how many diabetics we have in this country, what drugs they are taking, who is following, how many TB patients we have. You need big data analytics there. Next one. Next one, this is this COVID story. Now, wearable technology is another. Can you have wearable belts where, we can monitor fetal health? The lady can do that. Continuous monitoring of ECG, environmental sensor for asthmatics. Next one. Way for next one. Next. Gene therapy is one other area, two step, what is it? One step forward, two step backwards. I've been following this area for the last 30 years. But more recently, looks gene therapy may have consequences in some of these blood-borne disorders in particular. Next one. Similarly, stem cell therapy. I don't know where we are in stem cell therapy. Everybody makes mesenchymal stem cells. I know that. But this is another very, very promising area. Next one. This is one thing which you may not have heard, CAR T-cell therapy. This is very, very important. Now it has become a very huge chimeric antigen receptor T-cells. Whether the T-cells carrying the chimeric antigen, cancer antigen can be made, and then, if you can introduce in the blood circulation, it can identify the cancer cell and destroy the cell. Next one. And there's a very recent paper in Nature Biotech 2022, very interesting. You don't have to struggle hard, you simply inject the mRNA and you will get the protein, thing, and then you decorate it with antibody and then drive it. CD5 receptor is recognized. Next one. This is what I told you in the beginning, what is synthetic biology. Of course, this is a field developed by engineers and physicists, not biologists. Because this is something, because they always said, wire, transistor, valve, you provide, the machine runs continuously. Can the living cell run continuously? That was the question. Next one. So they started using, next one. They started using gene, protein, RNA instead of valves. All these there are being used. Next one. And this is where how you can do, you can at least 3 operons can be put there in a synthetic biology system. That means, you know, you in entire chromosomes can be introduced in a cell, and then you can get an entirely different product. I got interested because artemisinin was the first product to have been made by synthetic biology. Artemisinin is made by this plant called Artemisia annua. Now they made the yeast to produce artemisinin by putting the pathways. Next one, next one. There are challenges. Well, synthetic biology, US has a project called 1000 molecule project. Next one. There are many companies making it. Next one. Next one. This is the I'm coming to CRISPR-Cas. Next one. You know, next one. This is how CRISPR-Cas works. Next one. At least I should tell you this point, may not know. There are agriculture application at least is very, very important, is doable in India. And India has approved SDN1 and SDN2 levels, means specific nuclease 1, 2. Basically, the argument is these kind of mutations that are happening by the nucleus cleavage can be found as well in nature. That's how the government is permitting. And you know, this is something gene editing, great opportunity for India to move forward. Next one. And very recently, next one, this is the example. India has approved GM mustard. And it was something fighting going on for something, but so many people were opposing. Finally, the Prime Minister has taken the call. GM mustard is approved in India. So we have 2 technologies available. You have gene editing, you have at least gene modification, GMO. At least for a few cases, we can take it forward. Let's see. Next one. Regulatory issues, you should take, I told you SDN1, SDN2, GM mustard has been approved for commercialization. Next one. Next one, next one. Yeah, these are all where we can assume global leadership, vaccines, biotech, and you know, secondary agriculture. I think Dr. Deshpal Verma mentioned this thing. Service sector, you have to combine BT and IT, synthetic biology, nanobiology system. Next one. Finally, 2 minutes, I will finish. You know, these are all the career opportunities if students are looking for. Fermentation industry, agri-biotech, traditional medicine, marine biotechnology. Next one. And, you know, biopharmaceutical, diagnostic, devices, medical instrumentation. Next one. Sericulture, bioremediation. Next one. Of course, like me, you can survey public sector, or you can go abroad, become NRI, then you start advising us what we should be doing. That also is possible. Clinical trial, drug screening, all these are possible. What I'm saying is there are a lot of opportunities in biotech. Don't worry about your future. Next one. But the most important thing is to become a startup. You start your company and then give job to 10 others. This has happened. Next one. This is possible. Startups, you know. Next one. A few years ago, Bangalore was the only city in startup of the world. Next one. More recently, 2021, I find Delhi and Mumbai also have come in the list of countries where startup is growing on in a big way. Next one. Bioeconomy is $70 billion. The government has said $150 billion. We have startups of 4,000 something, hoping to be 5,000 startups. Next one. Next one, these are all revenues. Still biopharma is dominating. Next one. This is the biorack I told you, which is changed the culture, the entire, from idea to marketing, biorack is supporting. And I should compliment Dr. M.K. Ban. It was all Barnes' idea. He went abroad and he got this idea, started SPRE, became BIRAC. He is the originator. He unfortunately is no more with us. Next one. These are all the impact of BIRAC in statistics. Next one. Next one. Now these are all various examples of products which I have, I think you can see the malaria kit somewhere here, and many products have been listed. Then I told you 150 products have been done. Next one. I think, thank you. I think I'm done. Yeah. Sorry for, I don't know whether I have exceeded my time, but it's okay. Thank you so much. Yeah. But I